Am I audible? Yes. Am I audible? Hey students, am I audible? Hello? Good evening, good evening, good evening guys. I hope I'm perfectly audible and visible to you all. Welcome to Unacademy NEET English. I am your biology educator Ambika Sharma and it is the day 30th of our game of NEET 2.0. And guys, this is the chapter that we are going to discuss today. And after completing today's lecture, you know that, right, we'll master plant physiology. What say guys? Hannah, we'll master plant physiology. We have already completed plant growth and development, photosynthesis, and today the respiration in plants. And this week on Friday, we will complete neural control and coordination. So basically in this week, we'll complete the most scoring, the most important chapters of our NEET syllabus, isn't it? Isn't it? The plant physiology and the human physiology. Right? The plant physiology and the human physiology. So hope you guys enjoyed the classes. Hope you people are making the notes and hope you guys are practicing the questions as well. Right? We'll plan something. We'll bring the question practice sessions as well. Don't worry about that. We are going to take care of you. Okay? Don't worry about that. We are going to take care of you. And if you people are asking me my plan that what are we doing? What are we going to do after this plant physiology and human physiology thing, then guys, let's move to the cell cycle or biomolecules, or obviously we can even go for the diversity. So you people need to decide it, right? I will ask you this thing on the Telegram as well, and even after the lecture in the comment section, you people can tell me what do you want to start next, this or this. It's totally up to you. It's totally up to you. What say guys? It's totally up to you. So before proceeding, right, please let me know. Are you excited for the today's class? Are you excited for the today's class? If it is a yes, I want to see some fire emojis in the chat section and quickly hit the like button. If you're new to our channel, subscribe to our channel guys and share the session link with your friends and tell them ma'am has started the class. Right, ma'am has started the class. And if you are asking me the duration, I don't think that it will exceed 5 or 6 hours. Right, in 5 or 6 hours we can finish it with break, of course, with break. Okay, and now one more exciting thing for all of you, and that is this, right, this. So tomorrow at 6 p.m. right, you have to visit our channel, you have to click on this link and you will be directed for this amazing surprise. The teachers, right, your amazing educators and toppers, all of them are going to give you the tips as well. Just wait for it. So tomorrow sharp at 6 p.m. make sure you click here, notify me, right? Okay, chal. So let's get started. So what is the name of the chapter? The name of the chapter is Respiration in Plants. So I want to see the definition of respiration in the chat section quickly. Right, this is our chapter name, that is Respiration in Plants. Right, Respiration in Plants. So I want to see the definition in the chat section, guys, quick. And share the session link with your friends, quick. Share the session link with your friends. Tell them that ma'am has started the class. Quick, go for it. Go for it. Yes. Go for it. Plants need oxygen, breakdown of glucose, that's it, that's all, nothing else. Yes, do let me know in the chat section. I'm waiting guys. The session should be interactive. Acha, basic need of living being, okay? Basic need of living being, okay? Breaking down of glucose, okay. See, we are talking about the respiration in plants. In general, firstly, I'll start with the basic thing and then we will write the proper definition. Even in the case of plants, you know that plants will go for the photosynthesis, right? Plants, they do what? They do the photosynthesis. Don't mind me saying that, right? One of my students asked me this, that plants in photosynthesis, they make the ATP. Then why do they need the respiration? Why do they need to generate the energy, right? Even, yes, these kind of questions also I'm receiving. Okay, so that's why I'm explaining this. Listen to me. Guys, you know that plants, they also go for the photosynthesis. Photon, light. Synthesis means formation. So when we are saying photosynthesis, in the formation, in the presence of light, there is the formation. There is the formation of glucose. We know it very well, right? We know it very well. So in that photosynthesis, what is going to happen are plants, right? They are going to use the inorganic raw material like carbon dioxide, H2O, and in the presence of sunlight, right? In the presence of the pigment, your chlorophyll, you know it very well. It is going to give us the sugar, right? It is going to give us the sugar. Okay, the carbohydrates, right? Basically, the sucrose will be translocated in the phloem. Basically, we know the starch will form. This is what we know from the photosynthesis. And we know we all are dependent on plants. We need our food from the plants, isn't it? We are heterotrophic, plants are autotrophic, they are making the food and we are also dependent on them. We know it, right? Isn't it? We know it, right? So, when we talk about this photosynthesis, you know that we have covered that the light reaction is there, the dark reaction is. Don't get confused, ki, madam, this lecture is on respiration. Why are you talking about the photosynthesis? I'm trying to link the things. Okay, I'm trying to link the things and hope, and I'm trying my best to make it easy for you all. Okay. So the thing is the light reaction and the dark reaction. So guys, when we talk about the light reaction, in the case of light reaction, you know that obviously light is trapped. Yes, madam. Who's going to trap it? The pigments, the chlorophyll. So this light reaction is there in the thylakoid membrane, right? Because the protein complexes are present there, and from the light reaction, ATP, NADPH, right, and oxygen will evolve. This is what we get from the light reaction. ATP, NADPH. What is this NADPH? Nicotinamide adenine dinucleotide phosphate. Nicotinamide adenine dinucleotide phosphate, and the oxygen will be released. Now, when you talk about the dark reaction, it is there in the stroma. And we know that the word dark is misnomer here. What is the meaning of being a misnomer term? Matlab dark here, yeah, dark reaction doesn't mean that this reaction will take place in the darkness. No, it's not like that. It is directly, it is not, it doesn't need the light directly, but indirectly it is dependent on light. Indirectly it is dependent on light. So the word dark is misnomer here, right? So the word dark is misnomer here. Are you getting my point? Are you getting my point? So dark reaction is the word. It will be there in the stroma, and here your carbon dioxide will be reduced, right? It will be reduced to the sugar, right? To the glucose, to the sugar. This is what we know, right? So where this process is taking place? This process will be there in the chloroplast. Okay, this process is there in the chloroplast, right, bache? Right? Yes, the matrix of chloroplast is basically known as the stroma. And we know that the chloroplast is also a semi-autonomous organelle having the circular DNA, my dear students, and the 70S ribosomes, isn't it? These are the thylakoids, fret lamella. I'm not going to discuss it in detail, but I hope this part is clear to you. So this is about the photosynthesis where the oxygen will be released. But now, in the case of plants, when you are talking about the respiration, when you are talking about what? When you are talking about the respiration, right? In the respiration, what is going to happen? In the respiration, there is the breakdown of carbon-carbon bond. That plant is working, but that plant also needs energy. Right? Plants, they also require energy, right? They also need to break the glucose so that they can get the energy, so that they can do their activities now. Even in our case also, what is this respiration? It is the breaking down of carbon-carbon bond. It is the breaking down of carbon-carbon bond. We have to break that carbon-carbon bond of that sugars. Why? Because we want energy. And how are we going to break that carbon-carbon bond? We are going to break that carbon-carbon bond in multi steps, in many steps. right? There will be a process, a multi-step process in which we will break the carbon-carbon bond so that we can get the energy from it. It's not like that in one go you are going to break that carbohydrates, you're going to break the glucose, and then we will get the energy. No, in multiple steps, in many steps, under the obviously under the enzymes control, like enzymes are required, right? So basically in respiration, carbon-carbon bond we have to break, right? And that too in many steps, and for each step we need the enzymes, right bache? And then we will trap that energy, right? We are going to trap that energy, isn't it, guys? In the form of bonds, the ATP formation will be there. This is what respiration is. So in the case of photosynthesis, plant was making the ATP, NADPH and O2 here. That will be used here in the dark reaction. So ultimately you know that what is the purpose of photosynthesis. But when it is a respiration, the energy need to be utilized. Okay, the energy need to be utilized. I will relate it here. This part I'm going to relate with the breathing and exchange of gases as well. So if you people remember that particular chapter, right? You know now we started our marathons from this breathing and exchange of gases. Breathing and exchange of gases. If you people remember this, so here also we talk about the external respiration. We also talk about the external respiration. We talk about the internal respiration, if you people remember, and ultimately here we also discuss the cellular respiration. Remember, we also discussed the cellular respiration. So basically at the level of cell, at the level of cell also, what is going to happen? Like you are talking about a single cell, the glucose breakdown will be there and the energy will be produced, right? So here in this chapter, our story will revolve around this, the cellular respiration. Is that clear? Are you getting the context here, right? Are you people getting the context here, that what are we going to read and how important it is? So if you are not getting it, then it is your problem, because I have already explained it, nahin? Chalo. So external respiration, internal, and the cellular respiration. So at the level of cell, we will see the breakdown of glucose in many steps. Then further aerobic respiration, anaerobic respiration, fermentation, everything we will discuss here in this particular chapter. Okay, and here firstly let me write the definition and then we will go, right? So here there is the breakdown of glucose. What is there here? That there is the breakdown of glucose. So if there is a breakdown, what kind of process it is going to be? What kind of process it is going to be? Yes, there is the breakdown. So what kind of process it is going to be? Tell me. Tell me. Yes? Tell me. So let me write the definition from the NCERT. Till then you can just write it. So as I said, the breaking of C-C bond, matlab carbon-carbon bond. What are we doing? We are breaking carbon-carbon bond, right? Carbon-carbon bond of complex compounds. Actually, you are talking about the organic compounds here, right? What are we talking? We are talking about the organic compounds here. So breaking of carbon-carbon bond of complex compound through the oxidation of cells, through the, yes bache, how? Through the oxidation within the cells, leading to the release of energy, isn't it? Yes. So what is happening? The breaking of carbon-carbon bond of complex compounds. You can also mention organic compounds here, through the oxidation within the cells, leading to the release of energy and trapping of that energy, leading to the release of considerable amount of energy and trapping that energy to form ATP is basically the respiration. Right, bache, is basically the respiration. And to be very specific, it is that cellular respiration. Are you getting my point? It is that cellular respiration. So what this definition is saying, we have to break that carbon-carbon bond, that carbon-carbon single bond, obviously in between the sugars, right? Obviously in between the sugars. Are you getting my point? Are you getting my point? Yes. Are you getting my point? Yes. Okay, so right, so oxidation within the cells leading to the release of considerable amount of energy. So basically you are breaking that bond, right? The energy will be released, and the energy that is released, we are using that energy, we are trapping that energy in the form of bonds. We are making the ATP. We are making adenosine triphosphate. What is this ATP? What is this ATP? It is, the definition is adenosine triphosphate. The definition is adenosine triphosphate, right? So this is what we are going to do in the respiration. And the points that you need to take care, that it is a multi-step process, right? It's not going to take place in a single step. It is a multi-step process. Yes, which is Soumya, right? So what is it? It is a multi-step process. Okay, bache? So energy will not be released in a single step. Okay, okay, right? So, and why is it so? Why are we saying that it is a multi-step process? Because bache, if energy will release in a single step, na, that will be the heat energy, and that is not something that we want here, right? We have to trap that energy so that we can synthesize the ATP, right? So that we can synthesize the ATP. Okay, bache? Okay, okay, okay. So the energy that will be released in steps, okay, in steps, we can trap that as a chemical energy, as I said. Okay, we can trap that as a chemical energy, as I said. So any doubt? Yes, any doubt? Tell me, any doubt? Okay, so it's a multi-step process, right? It's a multi-step process controlled by many enzymes. Okay, it's a multi-step process which is controlled by many enzymes. Okay, done, bache? So ultimately we have to synthesize. So we don't want that energy to get converted into the heat. We want to trap that in the form of that chemicals, in the form of that ATP. So now, because there is a breakdown of glucose, what is happening here? Here there is a breakdown. Breakdown of what? There is a breakdown of glucose. So obviously if there is a breakdown, we should use the word, it is a catabolic process, yes or no? We are breaking the bond here, right? What are we doing? We are breaking the bond here. So don't you think we have to use this word, it is a catabolic process? Yes, don't you think we have to use the word, it is a catabolic process? Yes, it is a catabolic process because we are breaking that bond. We are breaking that bond. The catabolism is there, the energy, right? You are breaking something. Remember, kata, cut, breakdown. Anabolism is the build-up. But here, by the end of this chapter, we will say that respiration is amphibolic. Right? By the end of this chapter, you people are going to read that respiration is amphibolic. So what is the meaning of amphibolic? Amphibolic means, amphi means dual. Amphi means dual, means both. Both means here, when you are saying that a process is amphibolic, you will see it is catabolic as well as anabolic. So kata, as I said, means cut, means breakdown. Anabolism means build up, right? Anabolism means build up. Is that clear? Is that clear? Right. So breaking of carbon-carbon bond of complex compounds, right, through the oxidation within the cell, leading to the release of considerable amount of energy that will be used to synthesize the ATP is called the cellular respiration. Is that clear? The basics clear here? No doubt here there is a breakdown of the carbon-carbon bond, but by the end you will understand that overall respiration is amphibolic. Right? Overall respiration is amphibolic. So if it is a clear, want to see some fire emojis in the chat section, guys? Quick. And here, that's what you are going to see, right? We will talk about the mitochondria here. It is the mitochondria which is the powerhouse of the cell, right? It is something very simple and we know it, right? Even in our class 9th, we discussed that mitochondria is the powerhouse of cell. Why? Because it will form ATP. And even your Vizim sir, who says that physiology and psychology is same, even he knows that mitochondria is the powerhouse of cell. So here our story will revolve around mitochondria. Okay? Here our story will revolve around mitochondria. So this is the chapter. Let's start with the basic introduction part. So now you tell me, in the respiration, energy will be released. So is it the word that I can use here? Is it the word that I can use here? Like in the photosynthesis, it was anabolism, build-up was there. Here, there is the breakdown. There is the breakdown. The energy will be released. So here, let's not forget the NCERT. So you know that all of us breathe to live, right? And it is important. It is important. We need that gases. We need that oxygen. Why? Because that oxygen is going to oxidize the glucose, and that glucose is going to give me the energy. Why do we need the food? Because it is a fuel for our body, right? Because of that only, because it will give us the energy. It is the fuel of our body. It is going to give us the energy, and that energy is required for our functioning, for our proper functioning. So all of us breathe to live, but why breathing is so important, right? You know that we need energy for carrying out our daily life activities, and be it the absorption, the transport, the movement, whatever it is, we need the energy for that, right? We need the energy for that. So that energy is coming from the food, and how that food is giving us the energy? You know that food, right? Just like carbs, proteins, fats, they are present. They are present. Right, in our food, so we have fibers, we have carbs, we have proteins, we have fats, we have that things, right? But when it comes to the energy, respiratory substrate is the carbohydrate, because they give the instant energy. The same thing we will discuss here in this chapter as well, don't worry. Okay, we will discuss that here in this chapter as well. So whatever we are doing, we need the energy for that, and food will give us the energy. And how that food will give us the energy, that is what we need to discuss here. Okay, so they are saying the process of breathing is very much connected to the process of release of energy from the food. Okay, bache, so all the energy which is required for the life processes is obtained from the oxidation of some macromolecules, and we call it as food. So when we are saying macromolecules means their molecular weight is above 10,000 Dalton, right? Their molecular weight is above 10,000 Daltons. Are you getting my point? Their molecular weight is above 10,000 Daltons, right? Bache, जैसे your carbohydrates, polysaccharides, they are what? They are the polymers. Proteins, they are the polymers of amino acids, right? Bache, nucleic acids, they are the polymers of nucleotides. Fats, they are not literally the macromolecules, they are micromolecules. But you know that story, na? Retentate, filtrate, we'll discuss that in biomolecule. But as of now, when we are saying macromolecules, the compounds having molecular weight above 10,000 Dalton. Okay, bache. So only green plants and cyanobacteria. So cyanobacteria means, yes students, cyanobacteria means blue-green algae, right? Blue-green algae, photosynthetic bacteria it is. So green plants and these photosynthetic bacteria, cyanobacteria, right? These photosynthetic bacteria, cyanobacteria, that will do oxygenic photosynthesis, that will release oxygen, right? They prepare their own food by the process of photosynthesis. They trap energy, convert it into the chemical energy, and that chemical energy is stored in the bonds of carbohydrate like glucose, sucrose and starch. Okay, bache? Okay, yes, is that clear? Is that clear? Right? So the part, you know that in the case of plants, what do you think? The photosynthesis will occur in which part? The photosynthesis will occur in which part? Yes, tell me. In the plants, the photosynthesis will occur in which part? Arey yaar, in the leaves. Some answers are actually simple. In the leaves, of course, right? It will be there in the leaves, in the green part, right? Basically, in the green part, in the leaves, it will be done. It will be done. But there are non-green parts also there in the plant. And there, the respiration will occur. What will occur? The respiration will occur. So this is what given in NCERT, that not all cells, tissue and organs, they photosynthesize. The cells containing chloroplast, right, which is, and that are most often located in the superficial layers, they will carry out the photosynthesis. Clear, bache? So that non-green part, here we will have the food for the oxidation. So food has to be translocated to all non-green parts. So animals are heterotrophic, they are dependent on plants. So these are very basic things, right? These are very basic things that they are explaining, that we are dependent on plants, right? But bache, when we use the word saprophytes, you know na, saprophytes, they are the one that feed on dead and decaying organic matter. The one that feed on dead and decaying, right? Dead and decaying organic matter. The one that feed on dead and decaying organic matter. What is that? They are the saprophytes, right? And when it comes to the saprophytes, we have fungi, even the bacteria. Okay, bache. So now here in the NCERT is saying here in this chapter we are dealing with the cellular respiration or mechanism of breakdown of food within the cell that will release the energy and ATP synthesis will occur. So any doubt here? Any doubt here? Any doubt here? We are also covering the NCERT here in the lectures. Okay, any doubt here? Yes, guys, if there is no doubt, do let me know in the chat section. Quick. Yes? Chaliye. So photosynthesis takes place within the chloroplast, right? Right. Whereas in the eukaryotes, of course, breakdown of complex molecules will take place in the cytoplasm. What are they saying? These points are very important. And in the paper, usually we make mistake in such kind of statements. So what are, yes, bache, what are they saying? They are saying when it comes to the photosynthesis, that is in the chloroplast. I specified it. But when it comes to the breakdown of glucose, breakdown of these molecules, it will start in cytoplasm. It's not like that it will be completed there in cytoplasm. There are conditions. So it will start in the cytoplasm and then in the mitochondria, right? So as I said, the breaking of carbon-carbon bond of complex compound through oxidation within the cell leading to the release of considerable amount of energy is the respiration. So compounds that will be oxidized, bache, they are the respiratory substrate. Usually carbohydrates are oxidized to release the energy, but protein, even the fats and even organic acid can also be used, right, bache, under certain condition. Done, bache. So when it comes to the release of energy, it is a series of slow stepwise reactions controlled by the enzymes and it will trapped as the chemical energy. So everything is from the NCERT that we have covered. Is that clear? Is that clear? Yes. Right. So hence it is important to understand that the energy released by oxidation in respiration is not used directly, but it is used to synthesize ATP. Matlab, it's not like that you are breaking glucose, the energy is getting released and you are using it. No, the ATP synthesis will occur from that. Right? What is going to happen? The ATP synthesis will occur from that and that ATP we are using. Okay, bache? Okay, bache. So ATP is the energy currency because this is the ATP that we are using for our activities. Is that clear? Is that clear? Yes or no? Yes or no? So now they are asking us one thing. Do plants breathe? You tell me. Do plants breathe? You tell me. Do plants breathe? Quickly tell me. Then we have to jump to our main topic. This is just the introductory part. Do plants breathe? Yes. If it is a yes, then some green hearts in the chat. Quick. Then some green hearts in the chat. Quick. Exactly, right? So yes, plants do breathe. Now see, let's relate it with the basic metabolic need. You know that plants, they are fixed, they are localized, Am I right? They are fixed, they are localized, they are not going here and there, right? They are just fixed. Whatever activities they are going to do, matlab they are at this place and they'll do it. They are synthesizing their food by themselves as well. Now when you talk about the animals, okay, my animals, they don't look like animals, but they are. Okay, but when you talk about the animals, they'll move here and there, right? They'll move here and there. Okay, so when they are moving here and there, obviously their metabolic needs are more, right? Their metabolic needs are high. Their energy demand is basically high in the comparison of the plant. So yes, they both breathe. It's not like that they do not. But in the case of plants, because metabolic needs are less, right? So comparatively, they don't have, just in our case, we have a proper breathing system and the respiratory system and the circulatory system. Right, you know that our circulatory system, in our case, the circulatory system is there, the blood vascular system is there, we have the heart, the pumping organ, and that heart has to pump the blood. Okay, that heart has to pump the blood, and that blood will carry what? That blood is going to carry the oxygen to the different parts, isn't it? Isn't it? Yes or no? Right, yes or no? Yes or no? Done. So the point here is that you know that that blood will carry the oxygen and that will give to the different parts of our body, to each and every part of our body, right? But in the case of plants, you know that they don't have that heart which is going to pump their blood or something. They have the vascular tissue, they have the xylem and phloem, even different parts, they need the food. But here, comparative to the plant, the animal, the metabolic needs are less. So in the case of plants, if you remember, leaves, you know na, leaves are having what? Stomata. Remember stomata? Like in the epidermis. In the epidermis, you know that, right? The stomata is present. The stomata is present, if you remember this. In the dicots, bean-shaped cells, right? Surrounded by some subsidiary or accessory cells. So they are also the modification of the epidermal cell. Here also you will see the chloroplast, and if you remember, the inner wall of guard cells, it is thicker than the outer wall, and this pore opening is the stoma, singular, stomata, plural, right? Stoma, singular, stomata, plural. I hope you know that. Okay, I hope you know that. So that stomata is meant for the gaseous exchange, if you remember, right? That stomata is meant for the gaseous exchange, if you remember, right? So yes, the gaseous exchange is also there in the plants. Plus here on the bark, let's say it is having the secondary growth, it is having that woody part. So in the bark also, you know that there are some living cells, and that living cells are forming lenticels. Lenticels, by which the gaseous exchange is there. So in the case of plant, each and every part of the plant, they don't need a heart that will pump the blood, that will supply the gases, right? Here each and every part is basic, their basic needs are also less, and they are capable of doing it, right? They manage, they do that gaseous exchange, and they work accordingly. Just as I said in the case here, the bark, living cells are also there, right? Basically the lenticels, these structures are present when you talk about the bark, and through that the gaseous exchange will also take place, right bache? Right bache? So let's read it from the NCERT, right? That yes, plants, they require oxygen for respiration, right bache? And they also give out the CO2. Just sometimes during the night time, our elders, just in our village, it is very common that one should not sleep under the tree during night times. Why? No, they don't explain it, of course, but obviously during night time, photosynthesis is not going on, respiration is there, it is the carbon dioxide that will be released. It is the carbon dioxide that will be released. Okay? So plants, they have a system in place that will ensure the availability of oxygen, right bache? So plants, they do not have no specialized organs for gaseous exchange. But as I said, they have lenticels and they have stomata. They have lenticels and they have stomata. Is that clear? Is that clear? So each plant part take care of its own gaseous exchange needs, right? A very little transport of gases is required here. Plus, plants do not present great demands for gas exchange. So root, stem, leaves, they respire at the rates which are lower than the animals. So only during photosynthesis, large volume of gas will be exchanged. Each leaf is well adapted to take care of its own need. Okay, bache? Okay. Done? Yes. Right? So you know that plants, During photosynthesis, they itself release oxygen. So oxygen availability is not an issue in their case. And third point is the distance that gases must diffuse even in large bulky plant is not great. Okay, so each living cell in a plant is located close to the surface of the plant, right? This is true for leaves. You may ask, but what about thick woody stems? So as I said, in stems, the living cells are also organized in thin layers beneath the bark, right? And that openings are lenticels. I explained you already. Okay, I explained you already, and that is how the gaseous exchange will occur, right? That is how the gaseous exchange will occur. So cells in the interior, they are dead. They will just provide the mechanical support. So bark, which is a non-technical term, right? That bark. So in that bark, under that bark, basically, you will see that lenticels, these living cells are there, which will help in the gaseous exchange. So that is how in the case of stem, the gaseous exchange will be done. Is that clear? So most of that living part in the direct contact with the air. Okay. Okay, bache. So this is also facilitated by the loose packing of parenchyma cells in leaves, stem and roots, which provide an interconnected network of air spaces. So this thing is related to the anatomy. This thing is related to the anatomy. So when we discuss the structure of leaf even, right, especially in the case of dicots, I believe you all remember the palisade parenchyma is there, the spongy parenchyma is there. There also you see some air spaces, the cavities, right? Right, that will also trap that gases now. So that is how in the case of plants they fulfill their needs. Any doubt here? Any doubt here? So the complete combustion of glucose. When there is the complete combustion of glucose, the glucose is completely oxidized. So what will it give you? Carbon dioxide and water, obviously the energy. So what is this C6H12O6? This is glucose, right? So 6 oxygen molecules, so CO2, water and the energy will be released. Energy is coming out. It is not getting utilized, it is coming out. So definitely it is a catabolic process. But as I said, by the end of this chapter, we will see that respiration is amphibolic in nature. Okay, we will see that respiration is amphibolic in nature. Is that clear? Is that clear? Chaliye, the next thing I would like to discuss is that what are the differences? Okay, what are the differences in the respiration and combustion? Only one or two. Just we have used this word now, so that is why I'm asking. Yes? Tell me, respiration and combustion. Can you just differentiate that? Right? Right? Dekho, one thing is very simple, that it occurs inside living cell. And this is a non-cellular process, isn't it? This is a non-cellular process. Yes or no? One thing is same, but other than that, do you have any point? So, yes. But it is under biological control. Enzymes are there, right? It is under biological control. Enzymes are there. But the combustion, it is an uncontrolled process. You cannot control that thing. Both the things, they require oxygen. Here there is the oxidation of food, right? Right. And here basically the heat energy will be released. So it is an uncontrolled process. Uncontrolled process. It is a biochemical process. It is a biochemical process, but this is physico-chemical process, right? It is a kind of physico-chemical process, the respiration and combustion. And other than that, bache, more than basically here energy will be released in steps, right? 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 Energy is released in multiple steps, if you people remember. The energy is released in multiple steps, right? If you people remember. Okay, okay, but here, right? One step and the bonds will be broken simultaneously. All the bonds will be broken simultaneously, right, bache? So here, you can say that when it is the respiration, slightly more than 50%, slightly more than 50% of energy is released in the form of heat. I really want to highlight this point. Matlab in the case of respiration, no doubt we are saying that it is a multi-step process. In every step the energy will be released and we are using that energy for making ATP, right? But still even the energy will also be released in the form of heat, right bache? Matlab, but here in the combustion, most of the energy is released as heat. Okay, so that is how these two processes are different as well, right? That is how these two processes are different as well. Is that clear? So here the temperature will not, because in our body the cells are breaking down glucose, but that doesn't mean that they are raising the temperature of our body, right? But here you know that even the temperature will also become very high. So yes, you should know the differences in these two. So here you guys can see what NCERT is saying. NCERT is saying if this energy is to be useful to the cell, it should be able to utilize it to synthesize other molecule. So strategy is that plant cell uses the energy to catabolize, to break the glucose molecule in such that not all the liberated energy goes out as heat. That's why the word is respiration here, but not the combustion here, right Stella? That's why the word is respiration here, not the combustion. So we have to oxidize the glucose, but in several small steps to be just right, some steps to be just large enough such that energy released can be coupled to ATP synthesis. So energy released, why? Because we need to synthesize the ATP. Is that clear? We need to synthesize the ATP. Okay, bache? So oxygen is needed, carbon dioxide, water and energy will be released. But in the combustion reaction, it requires oxygen. But some cells live, right? Here, they have just given one line. The combustion reaction requires oxygen. Respiration also requires oxygen, but you know the difference in these two. Okay, bache? Okay, now they are saying there are some organisms that can even live without the oxygen. Now what will they do? Now actually it is our main chapter. Dekho, let's discuss it in the form of this, let's relate it with the evolution first. Do you know about evolution? Yes. Do you know about evolution? What do you know about evolution? Yes, bache? What do you know? What do you know about evolution? Dekho, when we talk about the evolution, we start the story with the Big Bang theory. I don't know whether you remember it or not, right? Don't worry, we will discuss it in the evolution chapter, right? We discuss about the Big Bang theory. What do we discuss? We talk about the Big Bang theory. Okay? Now, when we talk about the Big Bang theory, we talk about that explosion that we cannot measure in physical terms, this and that. You know that story. Matlab basically, a Big Bang will be there. We cannot measure that. The explosion is there, right? And that is not something that we can measure in physical terms, right? And you know that initially no atmosphere was there on our planet, right? I'm not discussing that nebula was there, planets, that's how formed. No, directly come to the point. So basically, initially, you know that when we talk about our planet, no atmosphere was there. Nothing, no life was there. With the time, what happened? We know that the life originated in water, right? With time, what happened? We know the most widely acceptable theory. We talk about the Oparin and Haldane theory, right? That is the naturalistic theory. That is the chemical theory. That says that firstly, there is the chemical evolution, then there is the biological evolution. That says that in the water, because temperature was already high there, right? that atoms, they reacted to form that, right? The atoms, they reacted, they form the molecules, then molecules form the elements, elements forms the compound, right? Do you remember that? Do you remember that? The element, they form the compound, right? The inorganic compound, that simple organic compound, complex organic compound, that is how the story started. And after that chemical evolution, that is chemogeny, right? Right, after that chemical evolution, that is chemogeny, the biological evolution, biogeny started, right? With the time, what happened, that organic compounds, they even start interacting, they started forming some protocells. They are not living. They are not living, right? They are cells. They are before life, right? They are not living, but they somehow resulted in the formation of the cell. And when we talk about the cells, bache, we discuss the prokaryotes first. I don't know whether you remember it or not, right? We talk about the prokaryotes first. So prokaryotes, P for prokaryotes, P, from this P, remember the primitive cells. So yes, initially prokaryotes, and that too, which are anaerobic. So what is the meaning of anaerobic? What is the meaning of anaerobic? Right? Matlab, without oxygen, these cells, they can survive without oxygen. They don't need oxygen for their metabolism. They can survive without oxygen. So initially, even when we talk about the evolution, we'll see the cells, they are anaerobic. They are anaerobic. They can survive without oxygen. Right? They don't need oxygen. And with time, with evolution, matlab, it's a time-taking process. It's not a one-day story, two-day story. So with the time, what happened, bache? With the time, what happened, right? The aerobic organisms, they evolved, right? Then they started the photosynthesis. Then they started the oxygenic photosynthesis. They started releasing the oxygen in the atmosphere. And slowly, the Earth atmosphere changed from reducing condition to the oxidizing one. Right, bache? So even initially, organisms, they were anaerobic. So obviously, they also need something now. Without oxygen, they are also, right? Without oxygen, right? Without oxygen, they were also surviving. And you know that it is metabolism, right? Which is making us living. This in biomolecule chapter in NCERT, that one line is there, no, which is saying like metabolism and living, we consider these things as synonyms. They are having the same meaning. If we are living in our cells, obviously the metabolism is going on, the chemical reactions are going on, right, bache? Right, bache? That is how we relate the things. I hope you know. Right, that is how we relate the things. I hope you know. So when it comes to the respiration also, right, basically what is there, right? We have the anaerobic organisms, we have the aerobic organisms. We have the anaerobic organisms, we have the aerobic organisms. But when we see it under the light of evolution, we'll see that even aerobic organisms, they evolved from anaerobic one. Can I say so? Yes, bache Namita. Yes, bache Sai, Harsha, Pooja, Tejaswini, Tej, Soumya. Tell me, guys. Rakev, focus here. Tell me, right? That is how it is. That is how it is. So basically your aerobic organisms, they somehow evolved from the anaerobic one. So when you talk about the respiration, right? When you talk about this thing, respiration, I really love this chapter and it's easy also. We start with the breakdown of glucose, right? We start the story with the breakdown of glucose. Right? How do we start our story? We start our story with breakdown of glucose. Right? And that breakdown of glucose, that first step of respiration is known as glycolysis. So glyco word is associated with the glucose. This lysis word is associated with the breakdown. Are you getting my point? This word is associated with the glucose. This lysis word is associated with the breakdown. Are you getting my point? Are you getting my point? So ultimately, there is the breakdown of glucose. Ultimately, there is the breakdown of glucose. So this is what glycolysis is. Breakdown of glucose. We are breaking the glucose, and that too in pyruvic acid. So two pyruvic acids we are going to get. 2 pyruvic acids. So this is the very first step of respiration. Let me try to make, you know, good notes, nice one. So when we are talking about the respiration, what are we saying? We are saying that when it is the first step, we are talking about the glycolysis, right? That is the breakdown of glucose. That is the breakdown of glucose. So this glucose, what is this glucose? It's a 6 carbon containing compound. Let's say 1, 2, 3, 4, 5, 6 carbon. Right? What is this glucose? That's how we write now 6C. What is the meaning here? 6 carbon containing compound. Right? 6 carbon containing compound. What's the formula? It's C6H12O6. It's C6H12O6. It's C6H12O6. So in the very first step, which is glycolysis, right, bache? Which is glycolysis, what is happening? We are breaking the glucose into 2, you can say pyruvic acid, or you can also say pyruvate. When we are saying pyruvate, we are talking about the ionic form. So 6 carbon containing compound. Why do we say 2 pyruvate? Because it is a 3 carbon containing compound. So from 6, you are getting 3. That's how it is. That is how it is. So this is the very first step, and it is oxygen independent. It is oxygen independent. What is the meaning of being oxygen independent? That it doesn't require oxygen. And where will it occur? The site is cytoplasm. It will take place in the cytoplasm. So this is the very first step of respiration. Very first step of respiration. Now when we talk about the pyruvate, this pyruvate is having 2 fates. This is the complete process, right? It's just like a flow chart that you people need to understand. Okay, then what is happening next, bache? This pyruvate is having 2 fates. Let's say if oxygen is present. If oxygen is present, matlab, what kind of conditions are we talking about? We are talking about the aerobic conditions. We are talking about the aerobic conditions. And if oxygen is absent, what kind of conditions are they? They are the anaerobic conditions. Okay, so guys, if aerobic conditions are available to this pyruvate will go for Krebs cycle. Before that, there is a link reaction. We will talk about it. And after Krebs cycle, further, there will be the electron transport chain. Further, there will be the electron transport chain. Matlab, if oxygen is available to that pyruvate, right, then it will enter in the Krebs cycle, obviously through a link reaction, and then the electron transport chain. Right? And you consider this topic as a difficult one, but actually it is not, right? Then you will go for the electron transport chain, and that electron transport chain will ultimately give us the ATP. And when anaerobic conditions are there, it will go for what? It will go for the fermentation. It will go for what? It will go for the fermentation. So even when we talk about the fermentation, students, right, we will talk about the lactic acid fermentation, lactate fermentation, and alcoholic fermentation, So we will talk about the lactic acid fermentation and the alcoholic fermentation. ठीक है जी? So the very first step is glycolysis, as I said, and it is oxygen independent. So when the organism is anaerobic, even if the organism is anaerobic or the aerobic one, they'll go for the glycolysis only. They'll go for the glycolysis first. So if it is aerobic, oxygen is available from this glucose, pyruvate will form, that will go for the Krebs cycle through link reaction, then electron transport chain. So basically, when oxygen is present, there is the complete oxidation of glucose. What is there? There is the complete oxidation of glucose, and that will result in the release of carbon dioxide, water, and the maximum energy that glucose can give us. But when there are such type of condition, when anaerobic conditions are there, there is the incomplete oxidation of glucose. Right, incomplete oxidation of glucose. So obviously a very less amount of energy, right, will be obtained. So this is something that we have to discuss here. This is something that we have to discuss in this particular chapter. But one thing that is very important, and from where we have to start it, is the glycolysis, that is common to both, whether it is an aerobic organism or an anaerobic organism. Is that clear? Is that clear? So it is also somewhere telling us about the evolution, right? It is also telling us about the evolution. Okay. So let's say that's a bacteria, and what kind of bacteria? Anaerobic bacteria. So how is it going to take the energy? In its cytoplasm, right? In its cytoplasm, what's going to happen? There is the breakdown of glucose in the pyruvate. So how many pyruvates will form from one glucose? 2 pyruvate or pyruvic acid. Right, bache? So that's why we are saying it is oxygen independent process. It doesn't need oxygen. And if there is error, if let's say obviously eukaryotic organism, right? So in the cytoplasm, glucose is giving us pyruvate, and that pyruvate will enter here in mitochondria, right? And obviously through link reaction, and further the complete oxidation will occur here. That is how it is. That is how it is. This is what we have to discuss. So let's start the story with the glycolysis, which is having another names also. Do you know about that names? Do you know about that names? Tell me. Do you know about that names? That is the word, right? So you can call it glycolysis, you can call it this Embden-Meyerhof-Paranis pathway and EMP pathway, whatever you want to say, right? Whatever you want to say. Is that clear? Is that clear? Yes. So glycose and lysis is the word that is given here. So let's discuss that. What is going to happen here? Okay. Done. So glycose associated with glucose, and lysis word is associated with breakdown here. So now when you are talking about it, now I hope you know it is common to both, right? I hope you will keep it in your mind. It is common to both. But as I said, the breakdown of glucose will be in the steps, right? It is not going to happen in one step. It is not going to happen in one step. It is a multi-step process. What's that? It is a multi-step process, right, bache? Right, bache? Yes, it's a multi-step process. So basically, when it comes to the glycolysis, you know, why do I repeat the same points? Because I want you people to just remember it always. So as I said, it's a multi-process. So total 10 steps are there. How many steps are there? 10 steps are there. In 10 steps, the glucose is going to give us 2 pyruvates, right? In 10 steps, the glucose is going to give us 2 pyruvates. Is that clear? Is that clear? Yes? Right? So it will be there in the 10th step. So before going forward, I'll tell you the kind of question that they can ask you in the paper. Dekho, it is a 10-step process. We have to remember all the 10 steps. And in my batch, I'm very happy that some of my students, right, they take it as a challenge. I challenge them that you have to remember the, right? I challenge them that you have to do what? That you have to remember the glycolysis, and they have done it. Right? And trust me, it's not difficult. For glycolysis, I do not have any trick because it's a 10-step process. But yeah, for Krebs cycle, for the electron transport chain, we have the tricks. right? For the Krebs cycle, for the electron transport chain, we have the tricks, but not here for the glycolysis. And trust me, it is very easy and interesting. Biochemistry is love. Trust me on that, right? And once you understand it, you just need some quick revision and everything will be on your fingertips. So as I said, what is the point here? They can ask you that glycolysis, where do it occur? Cytoplasm, presence of oxygen or absence? Absence. How many steps? 10 steps. Then in the paper, they can give you one particular reaction. Let's say glucose 6-phosphate will get converted into dash. That type of reaction. They can ask you about the enzyme, right? They can ask you about the enzyme as well. Right? It's not just that reaction. They can ask you about the enzyme. They can ask you about the reaction step. Plus, a very famous question here from the glycolysis is the substrate level phosphorylation. That how many steps of substrate level phosphorylation are there? Right? They can ask you what is substrate level phosphorylation. They can give you the reaction and they can ask you. In the photosynthesis, we talked about the photophosphorylation. Right? In the photosynthesis, what we discussed? We discussed about the photophosphorylation. Matlab, in the presence of light, there is the addition of phosphate to the ADP and the ATP formation is there, right? And the ATP formation is there. That is what we know. But here it is the substrate level phosphorylation, right? I'll explain it as well. So that type of questions will come in your paper. So are you ready to master this? Yes. Are you ready to master it? Tell me. Are you ready to master it? Yes. Okay, very good. So as I said, 10 steps. Okay? The very first step is the glucose. Yes, in the NCERT, the flow chart that they have given, right, it is not appropriate. And you know that in NEET from NEET 2024, we can learn one thing, that we have to focus on the concept, right? Not just the NCERT, the information should be correct. So I will show you something, this, right? This is from my favorite book, obviously, Stryer Biochemistry. So here, right, this flow chart will tell you everything correctly, right? We have to focus here that which steps are reversible, which steps are irreversible. So we will discuss it one by one. So are you ready? Are you ready? Are you ready? Firstly, let me write down the name of the compounds. Here, these are the terms that, these are the compounds that we will discuss. Glucose 6-phosphate. So for glucose 6-phosphate, I will use the short form G6P. Then here we will also talk about the fructose 6-phosphate. Right now, I am not saying that this will form this will form that. I'm just writing the compounds here. So, and in that sequence, we have to remember. So glucose 6-phosphate, fructose 6-phosphate, fructose 1,6-bis phosphate. Okay? Chalo, repeat it. Today we have to cram it, right? We have to cram it. Obviously, we are not going to understand the mechanism ki acha, this happened, that happened, right? Just let's cram it. Let's okay, do it in the class itself. So glucose will form glucose 6-phosphate, then fructose 6-phosphate. Glucose 6-phosphate, fructose 6-phosphate. That fructose 6-phosphate will give you fructose 1,6-bisphosphate, fructose 1,6-bisphosphate, right? So fructose 1,6-bisphosphate. Okay, why are we using the word bis here, right? Again, you know that, let's say 1, 2, 3, 4, 5, 6. You have a 6 carbon containing compound, right? If the phosphate is attached, 2 phosphates, if they are attached to the same carbon, you will use the word diphosphate. But if one phosphate is attached to one carbon, another phosphate is attached to another carbon, then we will use the word bisphosphate. And here bache, fructose 1,6-bisphosphate means one phosphate is attached to its first carbon, one phosphate is attached to its sixth carbon. So fructose 1,6-bisphosphate. What is the word here? Fructose 1,6-bisphosphate. Is that clear? It is fructose 1,6-bisphosphate. Understood? ठीक है जी, चलो 1-bisphosphate. Then बच्चे glyceraldehyde 3-phosphate. So the short form here is G3P or PGAL. Again, I'm going to relate it with the photosynthesis, right? G3P means glyceraldehyde 3-phosphate, means third carbon is having the phosphate here. Now, PGAL, G3P or PGAL is same. PGAL means phosphoglyceraldehyde. Sometimes we write it like this, 3-PGAL. Matlab third carbon is having the phosphate. Phosphoglyceraldehyde, right? It is phosphoglyceraldehyde, PGAL. ठीक है जी, PGAL. Now, do not confuse it with PGA. So, PGA means phosphoglyceric acid, right? PGA means phosphoglyceric acid. I specified the same thing in the C3 cycle also. ठीक है, G3P glyceraldehyde 3-phosphate, PGAL, G3P and PGL is same. Glyceraldehyde 3-phosphate, or you can say phosphoglyceraldehyde. Phosphoglyceraldehyde, right? And here it is PGA. PGA means phosphoglyceric acid. Functional group is different. Here it is aldehyde. Here it is acidic functional group, COOH. Here it is CHO. Are you getting my point? Do not get confused here, right? Then one more thing, dihydroxyacetone phosphate. Then what is it? Dihydroxyacetone phosphate या acetone 3-phosphate, whatever you want to, acetone phosphate. ठीक है, right बच्चे? Yes, tell me, is that clear? Is that clear? So now repeat it with me. Glucose 6-phosphate, fructose 6-phosphate, fructose 1,6-bisphosphate, glyceraldehyde 3-phosphate, and dihydroxyacetone 3-phosphate. Dihydroxyacetone 3-phosphate, right? Dihydroxyacetone 3-phosphate. Repeat it. Please repeat it, and trust me, it's easy to remember. It's easy to remember. In the NEET paper, you will get everything in front of you, right? You people are going to get everything in front of you. Okay, so you will be able to answer it. So let's revise it quickly. Let's revise it, right? So glucose 6-phosphate, fructose 6-phosphate, fructose 1,6-bisphosphate, glyceraldehyde 3-phosphate, dihydroxyacetone phosphate, right bache? Obviously the reactions are taking place. Glucose is forming glucose 6-phosphate. You are doing the phosphorylation here. What is phosphorylation? Addition of phosphate. From glucose 6-phosphate, you are making fructose 6-phosphate. Isomerization. Glucose 6-phosphate and fructose 6-phosphate, they are isomers. Isomerization is there. Again, there is the phosphorylation. Then there is the cleavage. These two are also the isomers. So that is how we will understand it. But I know there are many students, right, those who are not even clear with the compounds. So let's remember it, right? So guys, tell me, if you are ready to remember it, then I'll put my energy in this way. Otherwise, I have the charts. I'll show you, I'll explain you, and I'll leave. What do you want? You want me to explain it in this way so that we can just cram it with the class itself? Yes, if you want it in that way, do let me know in the chat section. Right, do let me know in the chat section, guys. Come on, quick. Yes, do it quickly. Good, very good. So then it is 1,3-bisphosphoglycerate, right? Even in the books, glyceric acid is written, yeah, phosphoric acid is written. Do not get confused. The ionic form, glycerate, ion. Carboxylic acid, yeah, carboxylate. It's like that. So 1,3-bisphosphoglycerate, then 3-phosphoglycerate, then 3 phosphoglycerate, then there will be 2 phosphoglycerate, then there will be phosphoenol pyruvate, and finally pyruvate. Finally pyruvate. That's what we have. So 1 2 3 4 5 6 ठीक है जी, done. ठीक है, so let's repeat glucose 6 phosphate, fructose 6 phosphate, fructose 1,6 bis phosphate, glyceraldehyde 3 phosphate, dihydroxy phosphate, then 1,3 bis phosphoglycerate, 2 phosphoglycerate, 3 phospho, 2 phospho, then phosphoenol pyruvate and pyruvate. Yes, quickly repeat it. Done? So tell me, is it easy? Is it easy or difficult? Is it easy or difficult? Is it easy or difficult? I think it's easy. I think it's easy. If we'll give it a try, we can remember that. Chaliye. So now I told you 10 steps are there. But we divide it into 2 parts. We divide the glycolysis into 2 parts. We say preparatory phase is there, and then what do we say? That there is the payoff phase. Preparatory phase is there, and then we say there is the payoff phase. So total 10 steps are there. Out of that, your 5 steps comes under this preparatory phase. 5 steps, they come under the payoff phase. Right? What is the meaning here? Ultimately, we are breaking the glucose. Why? Because we need to get the energy. Right? We have to make the ATP. But initially, in the starting of process, Bache Namita, we will see that we will use ATP. Right? We will see in the starting of the glycolysis, we will see that we use ATP. Right? And in the payoff phase, there is the synthesis of ATP. Okay, we form ATP. So that is why first 5 steps they are coming under the preparatory phase. Next 5 steps are coming under the payoff phase. Right? Right? Done? Yes? Done? So 5 steps are preparatory phase. Why do we say it is a preparatory phase? Because here in the glycolysis we are using the ATP. Next 5 steps, it is the payoff phase, matlab in which ATP formation is there. That is what you need to keep in your mind. Now let's go. Let's start. So the very first step is your glucose is forming glucose 6 phosphate. Now I'm not writing it completely. Okay, that is why initially I explained it. Okay, bache, that is why initially I explained it. So the very first step, right? Done, bache? Done, bache? So the very first step is the glucose will form glucose 6 phosphate. The very first step. And bache, remember, in the glycolysis, for each and every step, enzymes are required. And for that enzymes, right, it is for all the enzymes of the glycolysis, it is the magnesium which is the cofactor. Means magnesium is required. It is the cofactor for all the enzymes of glycolysis. Right? It is the cofactor for all the enzymes of glycolysis. Are you getting my point? It is the right? It is what? It is the cofactor for all the enzymes of the glycolysis. So in the very first step, in the glucose, we added the phosphate. So can I say it is the phosphorylation? Can I say it is the phosphorylation? Can I say it is the phosphorylation? Phosphorylation. What is phosphorylation? Addition of phosphate. Addition of phosphates. Yes, madam. We can say that. And this very first step is irreversible here. It is not reversible. We are not going to write it like this, ki glucose is giving us G6P. No, this is incorrect. The very first step is irreversible here, right? And which enzyme we can use here? Hexokinase. Kinase is associated with the phosphate, with the addition of phosphate. Kinase is associated with the phosphate, addition of phosphate. So it is hexokinase. Why hexo? Hexa means 6 carbon containing compound. And glucokinase is also used. Matlab both are correct. Both are correct. But glucokinase is used under certain condition. Hexokinase is used under certain condition. That is your homework, you will tell me. Matlab I'm not saying both are having same meaning. No, they are catalyzing the same step. When you are saying hexokinase, you are saying say hexa word, 6 carbon containing compound. Now you are specific, glucokinase it is, right? So they are having some different roles. I am not going to explain it here. I am expecting the answer in the chat section. And one more thing I would like to mention here, bache, and that is, just see, this is our channel. You know that in the video section, few days back I posted this video, reality check, a very amazing plan for all the students. Here in the comment section, I told you I will answer each and every question. So I'm not going to leave it. Just here, dekho. Some students are saying I cannot complete the entire chapter in one day, ma'am. What can we do? Right, I'm getting demotivated. One is saying, chalo, best video, thank you. Or thank you, mera bacha. I'm getting good comments also, and some questions are there. Just say my problem is missing interest, consistency, this and that. So I'm going to make a video where I will address all the comments of this particular video. So if you guys are also having some trouble, right, while preparing this NEET examination, right, in the preparation of this NEET examination, right, you guys can put it here in the comment section. Okay, I'll make a video and hopefully I'll post it by tomorrow or day after tomorrow, maximum day after tomorrow. You'll get that video. You can put it there. Same way, I also check the homework, right? So I'm asking you, you will tell me hexokinase, glucokinase. How are they different? It's interesting, yaar. Thoda level badhao, right? It's not always like it was given in NCERT, I know this. Show some interest in your subject, right? Show some interest in your subject. So if you have a problem, do let me know here in this particular video. So as I said, you can use anything, but there is a difference, right? You can use anything, but there is a difference. Okay? Done? Chali. So phosphorylation enzyme is there. It is irreversible. Now this glucose 6-phosphate. This is our first step. I'm telling you total 10 steps are there. Fructose 6-phosphate. I told you here. If still there is doubt, G6P, F6P. I'm telling you the short forms also. Fructose 6-phosphate. So they are isomers. So can I say that this reaction is the isomerization? Here the isomerization has been done. Yes, madam. And obviously magnesium is required. So glucose 6-phosphate, we are converting it into phosphate, fructose 6-phosphate. So obviously which enzyme will be used here? Isomerase. Glucose 6-phosphate, isomerase. So tell me, this part clear? This part clear? This part clear? Now, I would like to mention one more thing. You are adding the phosphate. What are we doing here? Right, this is glucose from the glucose, right? This is glucose. From the glucose, we are making glucose 6-phosphate. We are doing phosphorylation. We are adding the phosphate into the glucose. Trust me, this particular process will take time. Then you are going to love Krebs cycle. You are even going to love electron transport chain, right? Okay? Right? So basically you have added the phosphate in the glucose. So from where will you get that phosphate? Simple. In the very first step of glycolysis, we will use ATP. From that ATP, that phosphate will be added here to the glucose, and ATP will form ADP. And from adenosine triphosphate, adenosine diphosphate will form. Right? You understand this thing, then I am going to pinpoint some things here. Okay? Glucose to glucose 6-phosphate. The very first step, the phosphorylation, irreversible process. Right? Because we have added the phosphate here, we need the source of that phosphate. From here we are picking it up. Then there is the isomerization. Isomers will form. G6P to F6P. Now, from this F6P, again, see, this step is a reversible step. Right? Look at the arrow here. Look at the arrow here. Look at the direction, forward, backward, both. G6P is giving you F6P, isomers, fructose 6-phosphate. Now this fructose 6-phosphate is giving you fructose 1,6-bis phosphate. It is giving you fructose 1,6-bis phosphate. Yes, it is giving you fructose 1,6-bis phosphate. Again, we are adding the phosphate. Again, we are adding the phosphate. Yes or no? Again, we are adding the phosphate. Yes or no? Yes or no? Hanji, yes or no? Yes or no? Yes, madam. So from where you are getting that phosphate? From the ATP again. Here again you have used the ATP. Again you have used the ATP. Again you have added the phosphate. So again this step is phosphorylation. Phosphorylation, and the enzyme kinase will be there, but it is phosphofructokinase. What's the name of enzyme? Phosphofructokinase, which is sometimes also referred to as regulatory enzyme. I'll explain you the reason. Don't worry, I'll explain it again. Right? Let me finish the glycolysis, then I will give you the break to process the information. Okay? So right, kinase word is there. Again, I told you it is associated with phosphorylation. So from this word, from this substrate, your enzyme name is also associated, phosphofructokinase. Again, it is also irreversible. This step is also irreversible. Now, some students were confused. What is this preparatory phase? What is this payoff phase? Now clear? Now clear that the initial steps of the glycolysis, they involve the use of ATP. We are talking about the respiration. We are talking about the cellular respiration. The very first step in the cellular respiration, whether it is an aerobic organism or an anaerobic organism, it is the glycolysis. It is the glycolysis. It is the breakdown of glucose into the two pyruvates in 10 steps. So in that 10 steps, let's say you have the, I'm giving you a task. In first 5 steps, you have to make food. In next 5 steps, you have to eat food. Something like that. You got a task, you have to make the food, you have to eat the food. So in first 5 steps, you are making the food. In next 5 steps, you are eating the food. That is how in glycolysis 10 steps are there. First 5 steps are the preparatory phase, in which you are even using the glucose. You were supposed to form the glucose, but here, you were supposed to form the ATP, but you are using the ATP in first 5 steps. And in the next 5 steps, we will make ATP. That is what we are going to see here. So I am saying, here you have used the ATP. So in the paper, they can also ask you that in which steps of glycolysis ATP will be used. First or third step. First or third step may we use the ATP. Okay? And these 2 steps are irreversible also. Now, I'll ask you one thing. We go to the hospital. Jैसे doctors, they give us that saline, that glucose. I'm not getting into the details of that. I'm just trying to explain one thing to you. Right? Jैसे, let's say a person is weak. Right? A person is weak. That person is not well. A person is weak. We go to the hospital. During that condition, doctors, they put that solution in our body through IV. That solution in our body. Right? In general, we say, Arey, doctors, they are giving us the glucose or something. I hope you know that. Yes, I hope you know that. Hai na? Now see, understand this thing. A person is already weak. Right? The body is not so active. That person is sick. Then why it is important for doctors to give the glucose? Why can't they directly give the glucose 6-phosphate? Why in that solution glucose is there but not the glucose 6-phosphate? Can you tell me? Anybody in the class? Anybody in the class? Yes. So obviously students of my batch, they can explain it. They say in the very first step, our body is already weak. And in the very first step of glycolysis, right, we are using the ATP. So why can't we directly get G6P? Why can't we directly get G6P? Because this G6P is impermeable, right? It is impermeable, right? To plasma membrane. It cannot cross the plasma membrane. Right, bache? It is impermeable. It cannot cross the plasma membrane. Right, bache? So basically, why this first step is very important? It is a kind of locking step. Right? We are basically locking this glucose here. Right? This glucose is there in the cell. It can even go out, right? It is permeable. It can cross the plasma membrane. It can go out, but we do not want that. We have to continue this process. So this glucose will get converted into we have to convert this glucose into the glucose 6-phosphate, and this glucose 6-phosphate is impermeable to plasma membrane. It cannot cross the plasma membrane. And doctors, that is why they cannot directly, in that solution, that is why we cannot directly take the G6P. Is that clear? We cannot directly take the G6P. Is that clear? Sure. So that is something you need to keep in your mind. Okay? Now come to this part, because we have to save time as well. So as I said, glucose will give us glucose 6-phosphate. So the first step is irreversible one. Second step glucose is giving us fructose 6-phosphate. This is what you have to follow. I hope the screen is clear to you. If the screen is clear to you all, do let me know. And otherwise I'll do something, obviously. It's clear now? Chalo, very good. So in the first step, we are using the ATP. In the third step, so they can give you the reaction also, F6P, right, to F16P. This reaction is reversible or irreversible. That kind of questions can come in paper, right, bache? So for first step, it is the hexokinase used. In the second step, phosphohexose isomerase, you can simply write isomerase, and phosphofructokinase. Now, I told you it is a regulatory enzyme. I will explain you the reason, but not now. Let me finish this particular topic. Then I will explain you the reason. Okay? Chaliye. Now, fructose 1,6-bisphosphate. Dekho, if you ever get confused, see, it is making sense. Glucose, you are adding the phosphate. From where are you getting the phosphate? ATP. You are using the ATP here. Glucose to fructose 6-phosphate to fructose 6-phosphate. The simple isomerization is going on. Right, bache, isomerase enzyme. Phosphoglucoisomerase, yeah, you can say phosphohexose isomerase. Then fructose 6-phosphate to fructose 1,6-bisphosphate. You have added the phosphate, one extra phosphate we have added. So again, we have used the ATP, and the enzyme is phosphofructokinase, a regulatory enzyme. Now the next, so fourth step, I'm writing it here, I'm explaining it here, fructose 1,6-bisphosphate. Now, bache, the next step is cleavage. This is the fourth step. Glucose to glucose 6-phosphate. No. Fourth step. It is the cleavage. You are breaking it. How? Matlab, see, you have 1, 2, 3, 4, 5, 6. 6 carbon containing compound. First carbon is also having phosphate. Sixth carbon is also having phosphate. Now there will be the cleavage. there will be the cleavage. You are breaking it. And when it comes to the cleavage, the enzyme is aldolase. The enzyme is aldolase. The enzyme is aldolase. So it's a 6 carbon containing compound. Don't you think if you will break it, then it will be a 3 carbon containing compound? It is going to form a 3 carbon containing compound. Don't you think it will form a 3 carbon containing compound and phosphate will be attached there? So now when you talk about the fructose 1,6-bisphosphate, aldolase enzyme will cleave it into 2 compounds, 2 3 carbon containing compounds. One is your glyceraldehyde 3-phosphate, another is your dihydroxyacetone phosphate. And here also, this is the 5th step. This is the 5th step. These two are also the isomers. These two are also the isomers. I'm writing it properly so that you do not get confused. If fructose 1,6-bisphosphate, right, due to the aldolase, right, this is the 4th step. This is the 4th step. So what is happening? This is the glyceraldehyde. What's that? This is the glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. Done, bache? And in this is the 5th step. Again, this is the 5th step. And this is also reversible. And this is also, right? This is also reversible, na? Ki irreversible? These two both are reversible. Now, listen to me. Glyceraldehyde, the functional group is aldehyde here. Dihydroxyacetone, the functional group is ketone here. Pay attention, right? The functional group is aldehyde here, the functional group is ketone here, right, bache? And because these two are also the isomers, so here the enzyme used is again the isomerase, so it is triose phosphate isomerase. Triose phosphate isomerase. So firstly tell me, story up to this part is clear to you or not? Yes. Story up to this part is clear to you or not? Okay? Clear? Now one thing that you need to remember here. No doubt we are forming 2 compounds here. This reaction in forward, backward, both the direction. Basically what is happening? Here something very important is going to happen. And that is something which confuses the student. Right? That confuses the student. So listen to me. You have glyceraldehyde 3-phosphate. Aldehyde functional group, you have dihydroxyacetone 3-acetone phosphate, the ketone functional group. Actually, initially it will form like this, then your DHAP, because this is more stable, okay? Your DHAP, it gets converted into glyceraldehyde 3-phosphate. Matlab, jaise let's say, you are breaking it like this. Let's say this is a 6 carbon containing compound. We are breaking it. We have a 3 carbon containing compound. We have a 3 carbon containing compound. Right? And they are also the isomers. Right? They are also the isomers. But what is happening? The reaction is in such a way, or obviously it is related to the stability as well. Your this compound is getting converted into this. Matlab, this yellow will get converted into green. Matlab, initially when you are breaking it, you will get green and yellow, but after some time this yellow is getting converted into green. So what am I trying to explain here? We will form, after this cleavage, aldolase will do that cleavage, we will get glyceraldehyde 3-phosphate, we will get dihydroxyacetone phosphate, but that dihydroxyacetone phosphate will also get converted into glyceraldehyde 3-phosphate. So basically, what will we get? Can I write it like this? Can I write it like this? We are getting 2 glyceraldehyde 3-phosphate. Can I write it like this? We are getting 2 glyceraldehyde 3-phosphate. 2 glyceraldehyde 3-phosphate. Yes, that we are getting 2 glyceraldehyde 3-phosphate. Can we write it in this way or not? If it is a yes, do let me know in the chat. Tell me yes. Yes or no? Yes or no? Right? So G3P, DHAP will get converted into G3P. So can I say 2G3P? Can I say 2G3P? And again writing or showing 2G3P, I can simply put 2 here. Yes or no? Yes or no means now you have glyceraldehyde 3-phosphate. How many glyceraldehyde 3-phosphate? 2. Now the next step, the 6th step, now we have entered in payoff phase. You will get 1,3-bis phosphoglycerate here, right? It was G3P, 1-phosphate, but here 1,3-bis phosphoglycerate it is. 1,3-bis phosphoglycerate it is. So G3P is giving you 1,3-bis phosphoglycerate. And what is happening? Now from where we are getting this phosphate? 2PI. What is this PI? You are talking about the inorganic phosphate here. We are talking about the inorganic phosphate here. Inorganic phosphate. And why are we saying 2? Why are we putting 2 now? Because we have 2 glyceraldehyde 3-phosphate. If it is not clear, do let me know. I'll explain it again. Do let me know, I'll explain it again. Again, I'm telling you, bache, after the cleavage of fructose 1,6-bis phosphate, you will get glyceraldehyde 3-phosphate and you will get dihydroxyacetone phosphate. But your dihydroxyacetone phosphate will get converted into glyceraldehyde 3-phosphate itself. So you will get 2 glyceraldehyde 3-phosphate. So that is why I'm putting 2 here. That basically 2 glyceraldehyde 3-phosphates we have. 2 compounds we have. Right? Now, that's why here also we are putting 2. So instead of writing it like this, G3P, then 1,3-bis phosphoglycerate, G3P, and then 1,3-bis phosphoglycerate, I'm putting it like this, right? That basically 2 compounds are present here. So how? It is G3P, single phosphate. It is 1,3-bis phosphoglycerate. We have added 1 phosphate. From where are we getting this? From inorganic phosphate. This time we are not using ATP. This time we are adding inorganic phosphate here. And here your 2NAD, it is oxidized, will get converted to 2NADH. 2NADH. So what is this NAD? Bache, these are also the assimilatory part. So it is NAD plus means oxidized it is, right? And it is nicotine amide adenine dinucleotide. Nicotine amide adenine dinucleotide. It is nicotine amide adenine dinucleotide. Clear bache? Clear bache? It is nicotine amide adenine dinucleotide. It is oxidized and it will form NADH. It will form NADH, right bache? And that is nicotine amide adenine nucleotide, but this one is the reduced form, right? This one is the reduced form, and sometimes we write it like this, na, NADH plus H positive. NADH plus H positive. So obviously, you know that oxidized and reduced form it is, right? So it will get the electron as well as the proton, as well as the proton. So 2 NAD will give you 2 NADH, right? Like this, right, bache? So this is the 6th step here. So can I say that? Yes, G3P is forming 1,3-bis-phosphoglycerate. So somewhere phosphorylation is happening. Phosphorylation is also there. But along with that, what is happening? See, this one is getting reduced also. So when you talk about this particular step, let me show you here. This particular step is basically what, bache? The oxidation is also taking place and phosphorylation. Oxidation is also taking place and phosphorylation. Right, what is happening? Oxidation is also taking place and phosphorylation. So 2 inorganic phosphates are added here. 2 NAD are forming 2 NADH and H positive is released. Okay, bache? H positive is released. Right, bache? So glyceraldehyde 3-phosphate, right, is giving us 1,3-bis-phosphoglycerate. 1,3-bis-phosphoglycerate. Is that clear? So we can even write it 2 multiply, or you can even put it like this. You can even write it like this. No harm in that. No harm in that. Is that clear or not? And yes, again, this is also the reversible step. Reversible step. So can we just recall? Chaliye, let's do it. So we started the story with the glucose. Then glucose 6-phosphate. The reaction is irreversible. Here, the reaction is reversible. From glucose 6-phosphate, we are getting fructose 6-phosphate. Then, again, irreversible step. We are getting fructose 1,6-bis-phosphate. Now, this fructose 1,6-bis-phosphate, it is undergoing cleavage. Right? It is undergoing cleavage. And that too is also, obviously, I am not able to mark it. So, that is also a reversible step. So, I am getting G3P and dihydroxyacetone phosphate. But what is going to happen? This DHAP will also get converted to G3P. So now onwards, we have 2 G3Ps. We have 2 G3Ps. So this is the first step. This is the second step. This is the third step. This is the fourth step. This one is 5th step. But the conversion of DHAP to G3P is not any step. Right? Right, bache? It's related to stability and all. Now, this G3P, how many G3P do we have now? 2. Again, it is forming 1,3-bis-phosphoglycerate. It is forming 1,3-bis-phosphoglycerate. It is forming 1,3-bis-phosphoglycerate. So obviously, 2 inorganic phosphates are added here, right, bache? And your 2 NAD, they are reduced. 2 NAD, they are reduced. That is what you are going to get in your 6th step. So what is happening here? The oxidation, loss of electrons is also there, and phosphorylation is also there. Phosphorylation is also there. Are you getting my point? And which enzyme is going to do it? Which enzyme is going to do it? Of course, your dehydrogenase. Glyceraldehyde phosphate dehydrogenase. Glyceraldehyde phosphate dehydrogenase. Is that clear? Is that clear? Is that clear? Yes, tell me. Any doubt? Any doubt? Is it clear? Is it clear? So guys, if there is any doubt, so far 1 and 3, it is irreversible. What, right? Matlab, there are more steps also, but right now this is what I can say, these 2 steps are irreversible. Plus in step first and third, ATP, we have used the ATP, right bache? Right bache? So first step, phosphorylation, isomerization, phosphorylation, cleavage, then again isomerization is there, and here oxidation and phosphorylation. Any doubt? Any doubt? Now I'll ask you something, and that is common sense. That is common sense. See, if you will remember this now, this flow, right, then trust me, everything will be easy for you. Bache, your 1,3 bisphosphoglycerate. Now what do we have? We have 1,3 bisphosphoglycerate, right? Now bache, 1,3 bisphosphoglycerate is giving us 3 phosphoglycerate. This 1,3 bisphosphoglycerate is giving us 3 phosphoglycerate. This is the next step. This is the next step. It is the step number 7. It is the step number 7. It is the 7th step. Your 1,3 bisphosphoglycerate is giving us 3 phosphoglycerate, right? It is giving us 3 phosphoglycerate. 3 phosphoglycerate. Now it's very simple. Dekho, it is having 2 phosphate. It is having 1 phosphate. Can you see that? It is having 2 phosphate. It is having 1 phosphate. Can you see that? Can you see that? It is having 2 phosphate. It is having 1 phosphate. Can you see that? Yes, madam. So what about that extra 1 phosphate? Dekho, ADP, adenosine diphosphate, will form ATP here. Matlab in our 7th step, in the 7th step here, ADP will get converted to ATP. Now, how many 1,3 bisphosphoglycerate do we have? 1 or 2? How many 1,3 bisphosphoglycerate do we have, Shri Priya? 1 or 2? 1 or 2? 2, right? 2 and 2, 3 phosphoglycerate. So obviously the ATP formed here will also be 2 na. ATP formed here will also be 2 na. It's like bache dekho, G3P and G3P. You have 2 G3P. This G3P is giving you 1,3 bisphosphoglycerate. This G3P is giving you 3 phosphoglycerate. This G3P is giving you 2 phosphoglycerate. Then phosphoenol pyruvate and then pyruvate. Okay, this is also doing the same. This is also doing the same. So instead of writing it separately, what are we doing? What are we doing? We are just simply putting 2. Ki there are 2 compounds like this. That there are 2 compounds like this. Right, bache? There are 2 compounds like this. That is what we are saying. That is what we are saying. Right. So obviously, if here in this particular step, ADP is forming ATP. So obviously, because 2 times this reaction is taking place. So 2 from 2 ADP, you are getting 2 ATP. Is it clear? Is it clear? Is it clear? Is it clear? Yes? Is it clear? Is it clear? Right? So that's why we are writing it like this. Right? So here, this 7th step, the phosphorylation is there now. Addition of phosphate to this adenosine diphosphate. So it is the substrate level phosphorylation. So yes, you have to remember this reaction. So they can just simply give you this 1,3 bisphosphoglycerate is getting converted to 3 phosphoglycerate. What kind of reaction it is? It is substrate level phosphorylation. What's that? It is substrate level phosphorylation. What's that, guys? It is substrate level phosphorylation. Is that clear? Is that clear? Sure. Is that clear? Sure. Done? Done? Sure? Pakka? Why do we say it is the substrate level phosphorylation? Bache, see, basically ADP, adenosine diphosphate, is giving us adenosine triphosphate. 1,3 bisphosphoglycerate, it is the substrate here. From this substrate, we are taking the phosphate that will be added to the ADP so that it can give us ATP. Substrate level phosphorylation. Here we are not using the light. We are not using the light that is helping in the addition of phosphate. Here from the substrate itself, its phosphate will get transferred to the ADP. Right? Its phosphate will get transferred to the ADP. Can you see that? So substrate is releasing the energy for phosphorylation. Right? Substrate is releasing the energy for phosphorylation. From this substrate itself, the phosphate group, because it is releasing the energy, that phosphate group will be added to the ADP, we will get ATP. So this is the substrate level phosphorylation. So in the glycolysis, even in the Krebs cycle, wherever substrate level phosphorylation reactions are there, you have to remember that reactions, and trust me, we will do it. Trust me, we will do it. Is that clear? Is that clear? So this step is also irreversible. Okay? This step is also irreversible. So glyceraldehyde 3-phosphate is giving us 1,3-bisphosphoglycerate. So here in the 6th step, 2NADH will form, and we will, in electron transport chain, we will also convert it into ATP. Right? These are the assimilatory parts. So 1,3-bisphosphoglycerate will give you 3-phosphoglycerate. Again, 2 ATP will form. So this is the substrate level phosphorylation. Step number 7th will show us the substrate level phosphorylation, and step 10. Here you will see SLP, substrate level phosphorylation. Okay? Substrate level phosphorylation, and even this step is also irreversible. So the enzyme used here is, again, phosphorylation is there, it will be kinase. So it is phosphoglycerate kinase. Phosphoglycerate kinase. Right, it is phosphoglycerate kinase. Okay? Now, from the 3-phosphoglycerate, you are getting 2-phosphoglycerate. Don't you think it is a kind of rearrangement? Don't you think it is a kind of rearrangement? Yes? Yes. Don't you think that it is a kind of rearrangement? Yes. It is a kind of rearrangement. Matlab, your phosphate group was there at, say, 3rd carbon. We are shifting it to 2nd carbon. So it is simply the rearrangement. So whenever you will see the rearrangement, the enzyme used will be mutase. The enzyme used is mutase. Which enzyme is used, bache? Mutase. Mutase it is. Mutase it is. Rearrangement. Now, 2-phosphoglycerate is giving you phospho enol. En mane double bond, right? Alkene, ene, ol mane OH. So from 2-phosphoglycerate, you are getting phosphoenol pyruvate. You remember PEP, phosphoenol pyruvate, right? Look at the, so what kind of compound it is going to be? CH2. This is the compound. This is the compound. Are you getting my point? This is the compound, right? So what is happening here, right? 2H2O will release. This is the 9th step. It is the 9th step where the water will form. So phosphoglycerate will give you phosphoenol pyruvate. So the enzyme name is also easy, enolase. Enolase. Then phosphoenol pyruvate will form pyruvate. Now again, it is phosphoenol pyruvate. Now it is simply pyruvate. Where will that phosphate grow? Again here, the 10th step is also the step where ATP will form. It is also the substrate level phosphorylation. Substrate is providing the energy so that we can add the phosphate to the ADP and the ATP will form. So these are the 10 steps, bache. Pyruvate kinase is the enzyme that will be used. So this is the payoff phase, and this is the payoff phase, and this is the preparatory phase. Okay? 5 steps of preparatory phase, 5 steps of payoff phase. So yes, you should remember the reactions also. Sometimes they can give you phosphoenol pyruvate will convert it into dash. You should know what are they talking about. So let's discuss. So in the glycolysis, how many ATP we have used? 2 ATP. Am I right? 2 ATP. Steps. In which steps? Step number 1. Step number 3rd. So, can I tell you can you just tell me the reactions also? Can you just tell me the reactions also? Now we are quickly revising it also. First step, when glucose is getting converted to glucose 6-phosphate. 3rd step, where fructose 6-phosphate is getting converted to fructose 1,6-bisphosphate. ठीक है, चलिए. Now ATP formed 7th step, 10th step. So what are they? They are SLPs. What is the meaning of SLP? Substrate level phosphorylation. So the very first reaction is where your 1,3 bisphosphoglycerate is getting converted to 3-phosphoglycerate. Right, here we will see from ADP the ATP will form. This is the 7th step. This is the 7th step that you have to remember as such. Or 10th step mein what is there? Phosphoenol pyruvate is getting converted to pyruvate. Okay? This step is also where the ATP formation is there, and it is also the 10th step. So can I say that step number 1, 3, 7 and 10, 4 steps are irreversible? Can I say so? 4 steps are irreversible. Can I say so? Can I say so? Can I say so? So we have used 2 ATP. Ultimately, how many ATP is formed? Ultimately, how many ATP is formed? Tell me. We have used 2 ATP. But how many ATP is formed? Yes. How many ATP is formed? 4. So from glycolysis, what is the output? What is the output? Output is 2 pyruvate. Matlab what are we going to obtain? 2 pyruvate, 4 ATP, 2 NADH2, 2 water molecule. That is what we are going to get. But here ATP used 2. So net ATP formed is 4 minus 2. It will be 2 ATP only. Matlab it's like you are starting a business. You are starting a business. Matlab how can you understand this? Ki you are right. You are starting a business. You are like ki yaar, you please give me 2 lakh rupees. Right. Right. You give me 2 lakh rupees. Whenever I'll get something, I'll make profit, I'll give it to you. Just now let's say from your business, your profit is 4 lakh, but you, right, you have to give me my 2 lakhs back. So your net profit will be 2 lakhs only. Okay? Your net profit will be 2 lakhs only. It's like that. Initially you have used 2 ATP, but total production is 4. So out of 4 minus 2, it will be 2. 2 ATP and 2 NADH. And in the electron transport chain, we will ultimately see that this NADH2 will also give its electron and it will also form the ATP. So this is what we got. This is what we got. So yes, in the paper, that type of questions can come. This is the glycolysis. If there is any doubt, feel free to ask. After this, we will take a break. But let's read it from the NCERT first. Okay, let's read it from the NCERT first. So if you have any single doubt, do let me know. And yes, why I just said that this phosphofructokinase is the regulatory enzyme. That's what I just said now. It is the regulatory enzyme. It is sometimes also referred to as pacemaker of glycolysis. Pacemaker of glycolysis. So if it is the pacemaker, matlab it is something related to that rate and all. Okay, it is the pacemaker of glycolysis, right? Pacemaker of glycolysis. So what is the point here? What is the point here? Yes, tell me, what is the point here? Right, the point here is that this particular enzyme, it gets inhibited by ATP concentration. Matlab if there is high ATP concentration, it gets inhibited, but it gets stimulated by ADP and inorganic phosphate. So basically, if already ATP production is there, ATP concentration is more. So obviously we don't need the glycolysis, right? We don't need the glycolysis, right? But if ADP and inorganic phosphate is more, what will happen? The glycolysis is required. Right, bache? Right, bache? So that is why it is the regulatory enzyme. It is regulating this particular process. It is regulating this particular process. Yes or no? Yes or no? right? So this particular enzyme is an allosteric enzyme, allosterically activated by ATP, allosterically inhibited by ATP, right? So it is an allosteric enzyme. Okay, so inhibited by this enzyme is inhibited by ATP concentration and stimulated by ADP concentration. That's why it is the regulatory enzyme. Is that clear? So let's cover it from NCERT, then you will get a break. So term glycolysis originated from a Greek word. Glycose means sugar, and lysis means splitting, breakdown. So Gustav Embden, Otto Meyerhoff, J. Parnas, they have given this, right? So that is why we call it as EMP pathway also. That's why we can also call it EMP pathway, okay? In anaerobic organism, it is the only process in respiration, because after this in an anaerobic organism, either the fermentation will be there, otherwise this is the process by which they are getting the energy, right? And it is there in the cytoplasm, right? And it is there in all living organisms. So all living organisms, whether aerobic or anaerobic, they'll show the glycolysis. Any doubt? Any doubt? So here there is the partial oxidation of glucose, not the complete oxidation. Partial oxidation, and it will form us 2-pyruvate. It will give us 2-pyruvate. Is that clear? So in plants, this glucose is derived from sucrose. You remember in the plants, in the phloem, right? In the plants, in the phloem, what do we have? We have the sucrose. What do we have? We have the sucrose. So you know that it is basically what? Right? Sucrose it is. It's a non-reducing sugar. If you people remember, it's a non-reducing sugar. Right, bache? This sucrose is made up of glucose and fructose. And when it comes to the translocation, because this sucrose is non-reducing, that is why it is there in the phloem. So for the glycolysis, this sucrose will be converted to the glucose. So sucrose, it's a disaccharide, right? It is also the table sugar, cane sugar, inert sugar, right? Invert sugar, not inert sugar, invert sugar it is. So these are the different names here. So this sucrose is what? It's a disaccharide. What is it, bache? It is a disaccharide. It is made up of glucose and fructose. So it will get translocated like this. Okay, it will get translocated like this. So in plants, the glucose is derived from sucrose, which is the end product of photosynthesis, and from this storage carbohydrate. Okay, so sucrose is converted to glucose and fructose by the enzyme invertase, yeah, sucrase, and these two monosaccharide, they will enter the glycolytic pathway. Okay, so glucose and fructose, they are phosphorylated. They will give you glucose 6-phosphate. Hexokinase is used here. Then this glucose, fructose 6-phosphate. Glucose 6-phosphate will give you fructose 6-phosphate, and after that, bache, basically we know that glucose and fructose, its metabolism is same. So in glycolysis, a chain of 10 reaction under the control of different enzyme will occur, bache. Ultimately pyruvate will form. So this is what we have covered, right? So you have to see where will be the synthesis of ATP and where the ATP will be used. This is what NCERT is saying. So ATP is utilized at 2 steps. So that's why, because it is given in NCERT, that's why it's a repeated PYQ. So first, in the conversion of glucose to glucose 6-phosphate. Second, in the conversion of fructose 6-phosphate to fructose 1,6-bisphosphate. Again, I have explained it. That is how you get the PYQs. So fructose 1,6-bisphosphate will split into DHAP and PGAL. Right, we find that there is one step only where NADH2 is formed from NAD. So that is the 6th step where glyceraldehyde 3-phosphate is getting converted to 1,3-bisphosphoglycerate. Okay, so 2 redox equivalents are removed. Right, matlab 2 hydrogen atoms in the form of 2 hydrogen atoms, they are removed from PGL and transferred to the NAD. That's why we are saying oxidation and phosphorylation is taking place. So PGL is oxidized with inorganic phosphate to get converted to 1,3-bisphosphoglycerate. So this conversion, right, it is also an energy yielding process. The energy will be trapped for the formation of ATP. Matlab when your 1,3-bisphosphoglycerate is giving you 3-phosphoglycerate. So another ATP synthesis is during the conversion of phosphoenol pyruvate to pyruvic acid. So that's what you have to do. So can you then calculate how many ATP molecules are directly synthesized? We know that total 4, but the net gain is 2. Okay? Right? So pyruvic acid is the key product of glycolysis. So its fate depends upon the cellular need. Right? Its fate depends upon the cellular need. What is the meaning of this line? Right? What is the meaning of this line? The meaning of this line is, if that cell, if oxygen is available, pyruvate will go for Krebs cycle. Firstly, link reaction, then Krebs cycle. If oxygen is not available, it will go for the fermentation, right? Yes, sometimes it is the only process of giving energy to that organism. So cell needs will decide it, right? So that's it, that's all. Rest we'll discuss after break. So if you guys are having any doubt, do let me know. So let's meet at 8:45. So I am giving you a break of 30 minutes. Please have something, and then we will continue it for another 2 hours. Okay, then we will continue it for another 2 hours, and in that 2 hours, trust me, fermentation Krebs cycle will be completed. Then again I will give you a break, and then we will go for the electron transport chain and the remaining portion, which is not at all difficult. So I hope you enjoyed it so far, and trust me, it's easy, right? It might take some effort to remember the glycolysis, but in the case of Krebs cycle, I'll give you so many tricks. Okay, chaliye. Hello, hello, hello. I hope I'm perfectly audible. Hi. Hello. Bache, if you have a doubt which is not related to this chapter, I'll suggest you to ask it at the end and you can put that in the comment section. Okay, any doubt? Any doubt? Okay, so guys, let's go, let's start. So, done. So, glycolysis clear. Glycolysis clear. So, let's revise it quickly in 1 or 2 minutes. So, what are we going to, what we discussed there, that glucose will give us 2 pyruvate. Okay, and along with that 2 ATP we are going to get, 2 NADH, 2 we are going to get, and then the 2 water molecules will also be released. So let's not talk about the water, let's focus on these 2 things. Okay, let's focus on these 2 things. You might have seen a question, a PYQ, that total number of glucose, that total number of ATP that we get from the glycolysis, and sometimes they give the answer as 8 ATP, if you people remember. Actually, what is going to happen when we talk about the complete oxidation of glucose, right? When we talk about the complete oxidation of glucose, so we will see this NADH, right? Even it will pass its electron to electron transport chain and it will also form the ATP, right? So actually one NADH gives us 2.5 ATP, and because here we have 2 NADH, it will give us, yes, tell me, total 5 ATP. So 5 ATP from this, 2 ATP from this, it will be 7. It will be 7. Now you are like, madam, you were saying 8 ATP. So actually before that in the NCERT, it was given that 1 NADH2 gives us 3 ATP. So on the basis of that, 2 will give us 3 plus 3, that is 6, and 2 that formed during the glycolysis. So that is why we used to mark the answer 8. But actually it is not like that. It will be 7, and that we will learn with, right? That we will learn. Right, after completing this fermentation part, now we will start the Krebs cycle and then the electron transport chain, and then we will discuss it. Okay, chaliye. So now what is the fate of this pyruvate? We have how many pyruvates? We have 2 pyruvates. How many pyruvates? 2 pyruvate, right, bache? So what is the fate here? The fate of this pyruvate, as I said, it depends upon the cellular needs. It depends upon the cellular needs. Are you getting my point? It depends upon the cellular needs, on the needs of the cell. Okay, so firstly, let's say anaerobic conditions are available. Okay, oxygen is not available, then it will go for what? It will go for the fermentation. This pyruvate will go for the fermentation, and when we talk about the fermentation, we are going to talk about the alcoholic fermentation and the lactic acid fermentation. These are the 2 things that we have. What do we have here? We have the alcoholic fermentation, bache, and we have the, right, we have the alcoholic fermentation and we have the lactate fermentation, or lactic acid fermentation. Okay? So firstly, let's discuss the fermentation, then let's talk about the Krebs cycle. So any doubt here? Any doubt here? Right, bache? So when we are talking about the alcoholic fermentation means we are, that particular organism is giving us the alcohol. So guys, remember yeast. So this alcoholic fermentation is the most common type. It is the most common type, and we will see it in the fungi and in some higher plants. Okay? So when you are talking about the alcoholic fermentation, remember, even in the fermentation, there is no complete oxidation of the glucose. Glucose will not be completely oxidized, Tejaswini. Right, bache? Even here also, even if it is the alcoholic fermentation, the glucose will not be completely oxidized. Right, bache? Right, bache? So basically, what is going to happen here? In 2 steps, the step will be completed. Right, in 2 steps, this particular fermentation will be completed. right? And when you talk about the two steps, the trick is doctor. You want to be the doctor now, so we write it like this, DR. So the very first step will be decarboxylation, right बच्चे, मतलब CO2 will be released here, and the second step is the reduction. Second step is the reduction. So in the very first step, what is going to happen? Your 2-pyruvate, because we have 2 pyruvate molecules, that pyruvate will give you 2 acetaldehydes. Right, from that pyruvate we will get 2 acetaldehydes, and obviously CO2 will be released here. That's why it is the decarboxylation. From 2 pyruvate, Nimita, from 2 pyruvate we are getting 2 acetaldehyde, and the 2 CO2 molecules will be released. Right, bache? Right, bache? So from here, which enzyme is going to do that? Because it is a decarboxylation. So which enzyme is going to do that? Pyruvate decarboxylase. Can I say that? It is pyruvate decarboxylase. Yes, pyruvate decarboxylase. It is the enzyme that is going to do that. Right? What is the name of the enzyme? It is the pyruvate decarboxylase or pyruvic acid decarboxylase. Right, bache? Right. And other than that, when it comes to the cofactor, we talk about the TPPs and the magnesium ion. Again, very important. Now, what is going to happen to this acetaldehyde? Now, the reduction. The next step is of reduction. So, these 2 acetaldehyde, will give 2 ethyl alcohol, right? It will give 2 ethyl alcohol, and obviously the energy will be produced. So what I just said, I talked about the fungi here. So obviously in the case of yeast, bache, in the case of yeast, you know na, it is the baking or the brewing yeast, right? That alcohol production. That is why we use the yeast in the fermentation, the enzymes. In the as a brewer's yeast, right? Why? Because it can produce the ethyl alcohol, ethanol. Right, bache? So when you talk about the fermentation, obviously it is not going to give us the complete, it is not going to do the complete oxidation of the glucose. Obviously the complete energy will not be released. Very less amount of energy will be released. So there are two things, alcoholic fermentation and the lactate fermentation will be. And the most common type is alcoholic fermentation. So we talk about the yeast, bache, right? You know that yeast, right? It is used in the baking industry or in the brewing industry, right, bache? So jaise let's say when you are talking about that brewing industry, you know that from this alcoholic fermentation, even that carbon dioxide is getting released. So that puffed up appearance, right? Jaise let's say if you are talking about the dosa or any other thing, matlab basically when we want to have that puffed up appearance, even in the case of cheese also, right? Even in the case of cheese, if you know Swiss cheese is having that holes, why? CO2 production. That swelling of that dough, why? CO2 production. So that is how it is used. So there is a very amazing point related to the yeast as well. We will discuss it. So in fermentation, say by yeast, the incomplete oxidation of glucose will be there, bache, right? So alcoholic fermentation, it is there in the 2 steps, right? So pyruvic acid is converted to carbon dioxide, matlab carbon dioxide will release. That's why first step is decarboxylation, and these two are the products here, CO2 and ethanol. These two are the products of that alcoholic fermentation. Okay. So pyruvic acid decarboxylase and alcohol dehydrogenase will catalyze these reactions. Just here I said na, pyruvic acid or pyruvate decarboxylase. So second is alcohol dehydrogenase, right? Here this reaction is catalyzed by alcohol dehydrogenase. So yes, the name of the enzymes, they are very important. The name of the enzyme is very important. Clear, bache? Clear. So when we are converting, again here I am writing 2 pyruvate, or you can say that pyruvic acid to 2 acetaldehyde, 2 pyruvate to 2 acetaldehyde, right? And 2 CO2 are released. So obviously, which enzyme is there? It is pyruvic acid decarboxylase. So this enzyme name is telling us that decarboxylation is taking place. Understood? Decarboxylation is taking place. Understood? Or second step mein what is happening? The 2 acetaldehyde, right? That is giving us what? That is giving us 2 ethyl alcohol or ethanol. right bache, and the energy is released. So here what is the point? Here the alcohol dehydrogenase enzyme is there. I hope it is on your fingertips now. I hope you people are clear here. Any doubt? Yes bache, any doubt? Tell me, any doubt? Yes? Any doubt? Okay, chale. So other organisms like some bacteria, they also produce lactic acid from pyruvic acid, right? So now when you're talking about the lactic acid production, right? Lactic acid fermentation or lactic acid production. One second. Students, even in the chapter locomotion and movement, I told you about the lactic acid formation, if you remember. Just say when you talk about the muscles, right? When you talk about the muscles, you know that in the case of muscles, it is the myoglobin, that is the storing pigment that will store the oxygen. Hemoglobin, it's a quaternary protein. Myoglobin is a tertiary protein. Hemoglobin is basically helping in the transport of that oxygen. Myoglobin is associated with storing of that oxygen. Muscles are having the myoglobin. Now when we are doing excessive exercise or something, we get that muscle cramps. What are that muscle cramps? Actually, it is the lactic acid accumulation. It is actually what? It is actually the lactic acid accumulation. The lactic acid is getting accumulated. So that accumulation of lactic acid is basically the fermentation, right, bache? That lactic acid accumulation is what? It is basically the lactic acid accumulation is what? It is basically that muscle cramps that we experience. So what is happening? Muscles are doing excess of work, right? So the oxygen is not available there. Oxygen is unavailable in that case, right? Because let's say if you are doing strenuous exercise, you're working hard a lot. Let's say today itself you started running. We feel that cramps. Why? Because our muscles, they need the oxygen, they are not getting it. They need the energy, they are not getting it. They need the energy, they are not getting it. So when that is the case, our muscles will be like, chalo theek hai yaar, I'll go for the glycolysis. Glycolysis to definitely be there. So muscles from glycolysis, they will form pyruvate. Now what is going to happen? They need more energy, that pyruvate, and oxygen availability is also there. Matlab anaerobic conditions are there in muscle. Anaerobic conditions are there in muscle. So what is going to happen? That pyruvate will get converted into the lactate. So it's a one step. It is what? It is a one-step process. Jैसे alcoholic fermentation. Okay, alcoholic fermentation is a two-step process. Alcoholic fermentation is a two-step process, and moreover, what is going to happen when this second step is there? You will also see that NADH is also used. Right, this reduced NADH will get oxidized here. Right, it will get oxidized here. Right, so here in the case of this lactic acid fermentation, you will see there is only one step, and in that step the pyruvate, these two pyruvates, they will get converted into the lactate. They'll get converted into the lactate, and obviously the enzyme is lactic acid dehydrogenase. What is the name of the enzyme? It is the lactic acid dehydrogenase. Okay, it is the lactic acid dehydrogenase. So in our case, our muscles are going to show this fermentation, right bache? And you know that when it comes to the cofactor, here the zinc ion is required. Here the FMN, flavin mononucleotide, is required. Right, FMN is required, zinc ion is required. Last reaction may it is the magnesium ion. So 2 lactate will form, and here also the NADH2 will be used here. Is that clear? Is that clear? Matlab your 2 NADH. Wait, I'm writing it here. So we have 2 pyruvate. 2 pyruvate is given. We have 2 pyruvate plus so from this pyruvate we are getting lactate plus this. Okay, and obviously energy is released. So as I said, the enzyme name is this, and FMN, flavin mononucleotide, and zinc ion will be used here. Flavin mononucleotide and zinc ion will be used here. So this is what fermentation is. Clear bache? This is what fermentation is. Is that clear? Is that clear? Yes. Is that clear? Sure. Any doubt here? Pakka? Okay, so here you guys can see the enzyme pyruvic acid decarboxylase and alcohol dehydrogenase will catalyze these reaction, right? So some bacteria, they also produce lactic acid from the pyruvic acid, right? So I have given you the example of mussels, and even lactic acid producing bacteria is there, right bache? So in animal cells mein, muscles during exercise, when oxygen is inadequate, when it is not sufficient for cellular respiration, pyruvic acid is reduced to lactic acid by lactate dehydrogenase, and reducing agent is this. It will be reoxidized to NAD in both the processes. So that is what we have discussed. So Anamika, Pujashree, any doubt? I hope it is clear to you all. I hope it is clear to you all, right? So this is what fermentation is. Now let's move to the next slide here. So here in both lactic acid and alcoholic fermentation, not much energy is released. It is less than 7% of the energy in glucose is released, and not all of it is trapped as high energy bonds. So imagine less than 7% of the energy of glucose is released in fermentation, because it is not the complete oxidation of the glucose, right? And it will not be even trapped in the form of ATP. ठीक है बच्चे? So process is hazardous also. जैसे even the acid production, lactic acid or alcohol is produced. So how much will be the number of ATP? When one molecule of glucose is fermented to alcohol or lactic acid, how much ATP we are going to get? You just need to focus on the ATP of your glycolysis. That is 2 ATP. That is 2 ATP only. Right? That is 2 ATP only. Right? So look at this reaction. Look at this reaction. Ultimately, this is the pyruvate. So lactic acid, from pyruvate, when we are getting lactic acid, see, one arrow is there. Your NADH2, it is reoxidized to NAD. Okay, only one arrow, matlab one step it is. Here 2 arrows are there, you are talking about 2 steps. So ultimately the end product is ethanol ya ethyl alcohol plus CO2, right? So do not miss this. So in the paper they can just give you this flow chart and they can ask you what is present here. So you should know, here they have mentioned glyceraldehyde 3-phosphate, 3-phosphoglyceric acid, phosphoenol pyruvate, like this. So here this NADH2 has formed, and here it will be used if this is the fermentation. So very interesting point about the yeast, right? I always mention it when I teach microbes in human welfare, ki yeast poison itself to death. Yeast poison itself to death when the concentration of alcohol reaches about 13%. Means, let's say there is a culture, yeast cells are there, right? Yeast is a unicellular fungi, belongs to class Ascomycetes, isn't it? Isn't it? Isn't it? Right? Yeast is what? Yeast is a unicellular fungi. Yeast is what? Yeast is a unicellular fungi, and it belongs to the class Ascomycetes. Am I right here? Yes, bache. Am I right here? Yeast is what? It is a unicellular fungi that belongs to the class Ascomycetes. Okay? So let's say there is a culture. And there is a culture, right? We have provided all the substrate to the yeast, right? Yeast is feeding on it. Obviously it will reproduce, it will use all that enzymes there. So what's going to happen when in that particular culture, let's say if the alcohol concentration, right? Let's say if in that particular culture alcohol concentration, if it reaches about 13%, right? If it reaches about 13%, so yeast will poison itself to death. Matlab, if alcohol concentration is 13% there, the yeast will not be able to reproduce, right? It will die. It will die. Okay? So that is why, due to the undistilled beverages that we get, they are having less alcohol concentration. In distilled beverages, it is more. Right. So see, what then would be the maximum concentration of alcohol in beverages that are naturally fermented? Naturally fermented beverages are comparatively, they are having the less, they are having the less alcohol quantity in comparison to the distilled beverages. Like if you are comparing a beer and the whiskey is having more concentration of alcohol because it is because of the distillation. Okay, bache? Okay, chaliye. So this question you have to answer. We also discussed the same in the microbes in human welfare, that how do you think the alcoholic beverages of alcohol content greater than this concentration are obtained? Obviously distillation. Okay. Okay, okay. So less than 7% of the energy will be taken out, right? So you know that in eukaryotes, these steps take place within the mitochondria and this. So obviously fungi is also eukaryotic cell. I hope you know that. Fungus is also a eukaryotic cell. Now, we are talking about the aerobic organisms which are having high metabolic needs. In their case, what will be the fate of pyruvate? What will be the fate of pyruvate? You know that it will go for the complete oxidation, further oxidation. It will go for the Krebs cycle, it will go for the electron transport chain, but before that there will be the link reaction. That is the fate. That is the fate of the pyruvate. That is the fate of the pyruvate. So you remember when I was explaining you this particular pathway in the starting itself, you remember this flow chart. So glycolysis will give us the 2-pyruvic acid. So the cellular needs will decide. So cellular needs and that conditions will decide. Oxygen present, it will go for Krebs cycle and electron transport chain. But before that, there is a link reaction which is actually linking this glycolysis with the Krebs cycle. If anaerobic conditions are there, fermentation. Lactic acid fermentation and alcoholic fermentation will be there. Any doubt? Any doubt, guys? Any doubt? So if there is no doubt, I want to see some energy in the chat section. Right, bache, I want to see some energy in the chat section. Yes or no? I want to see the energy in the chat section. Yes or no? Yes or no? Yes or no? Very good, very good, very good. So this fermentation thing clear? Now we will talk about the Krebs cycle. But before this Krebs cycle thing, जैसे here, see, it is given na, aerobic respiration is the process. It is the process that leads to complete oxidation of organic substrate in the presence of oxygen and release of CO2, water and large amount of energy. Right? It is the process that leads to the complete oxidation, of organic substance in the presence of oxygen and CO2, water and large amount of energy will be released. Okay? So this type is most common in higher organism and that's what we have to discuss now. Okay? That is what we have to discuss now. Okay? Okay? So what are they explaining here? Chalo, before this I will explain it. So what's going to happen? Your pyruvate, we have 2 pyruvates. We have 2 pyruvates. Where are they present? They are present in the cytoplasm. Where are they present? They are present in the cytoplasm. Now, because it is the respiration, which organelle is going to play the role? It is your mitochondria. We will discuss the structure of mitochondria as well. Don't worry. We are going to discuss the structure of mitochondria as well. Mitochondria, the powerhouse of the cell. Right, the powerhouse of the cell. So right now I'll teach you some basic points related to it and one by one we will discuss it in detail. So when we are talking about the mitochondria, you know that it's a double membrane bound organelle. Right, it's a double membrane bound organelle. So you have outer membrane and you have inner membrane. You have outer membrane and you have inner membrane. Now in between these two, what do we have? We have the intermembrane space. Inter means in between the membrane space is there. So you have outer membrane, you have inner membrane. Firstly, let's talk about the mitochondria, then we will relate it here. Outer membrane, inner membrane, and in between these two membrane, intermembrane space is there. They are perimitochondrial space. Now, if you will see, the inner membrane is showing some enfoldings. We are calling it as cristae. We are calling it as cristae. What is it? Cristae. Now it is the mitochondria, but it is a semi-autonomous organelle. What kind of organelle it is? It's a semi-autonomous organelle. Semi means half. So actually what's the point? Bache, we know that plastid, mitochondria and prokaryotes, they share the similarity. Just like prokaryotes, even in mitochondria, we have the circular DNA and we have 70S ribosome. Okay? Yes, here in mitochondria also, we will see the circular DNA just like prokaryotes and the ribosome is 70S ribosome, right? 70S ribosome. So S stands for Swedberg unit here. S stands for Swedberg unit here or the sedimentation coefficient here. S stands for Swedberg unit or the sedimentation coefficient here. Are you getting my point? So we are saying it is a semi-autonomous organelle, right? right? So it is the circular DNA, 70S ribosome is there. S is the Swedberg unit, sedimentation coefficient it is. This is your what? This is your mitochondria. And here, this homogeneous, this substance here is what? It is the matrix. It is the matrix. These are the terms that I'm going to use here, and I don't want you people to not to understand that. Okay. So this is a double membrane bound organelle. It is the powerhouse of the cell. It is having the enfolding, the matrix, just like prokaryote, circular DNA, and 70S ribosome is there. So because it is having DNA, it is having the protein factory. It can make some protein. It is not completely dependent on nucleus, right? It can fulfill its needs also. It is also synthesizing some of the protein that it needs. Okay, and moreover, just like prokaryotes, it divides by fission, right? The mitochondria, your chloroplast, they can divide by fission. Okay, this is the mitochondria. Okay, this is what? This is the mitochondria. It is the matrix. So guys, this is the matrix. This here in the matrix, the Krebs cycle. Students, here in the matrix, the Krebs cycle will occur. It is the location of the Krebs cycle. It is the location of the Krebs cycle. Now, when we talk about the infoldings here, right? Just cristae. What will they do? They will increase the surface area. They will increase the surface area. Here in the cristae, you will see some structures, right? We will see some structures like this, right? We are going to see some structures like this. Right, so these are what? These are the oxysomes. These are what? These are the oxysomes, also known as ATP synthase. I hope you can correlate it with the photosynthesis also. It is also known as F0 F1 particle. F0 F1 particle. F0 F1 particle. Let me explain this. Let's say if this is your mitochondria, you are talking about the cristae here. So ultimately, it is also the phospholipid bilayer, nothing else. It is also the phospholipid bilayer, nothing else. Yes, nothing else. Right. So here also in this phospholipid bilayer, right? Here also in this phospholipid bilayer, here also in this phospholipid bilayer. What's the point? There is some structure like this, right? Matlab this part is facing the matrix. What is it? Bache, they see the portion which is traversing in the membrane, which is present in the membrane. This part is basically F0, right? Let's say this is the structure. This is the structure. So this part is the F0. It is the base. Right, and this part here, which includes the head plus stalk, it is the F1. It is the F1. It is the F1, and we are also calling it as what? We are also calling it. So this is the oxysome. What's that, bache? It is the oxysome. This is the oxysome. We need it for electron transport chain. So firstly, you people should understand the mitochondria structure. Then further, I will explain you everything. Okay? Then I will explain you everything further. Okay? This is the structure here. So this base, which is traversing there in the membrane, it is present there in the membrane. That is also a PYQ. Right? That is also a PYQ. So which part of this oxysome traverse the membrane, which is transmembrane, which is there in the membrane. So obviously it is the F0. It is the F0. Is that clear? And this part here, the base and the stalk is F1, which is there in the matrix. Which is there in the matrix. Here you will see it. Right? Like this. Okay, so the cristae, the number of cristae also, it depends upon the cell's need, right? Because they are increasing the surface area. And what do we have in this inner membrane? We are having many enzymes here, and that inner membrane is the place where the electron transport system is present. Namita, here in the inner membrane, electron transfer chain, transfer system is present. Now, transport system is present. This is the outer membrane. Now, when you talk about its outer membrane, it's quite permeable. This is the outer membrane, this is the inner membrane. But if we talk about their permeability, matlab how much they are allowing the things to move here and there. So outer membrane is more permeable. Yeah, it is considered as permeable, right? More permeable than inner membrane. It's considered as quite permeable, right? Why? Because it is having some, right, here in the outer membrane you will see that there are some proteins that are forming the kind of channels, kind of pores. There are some protein channels that are like pores. We call them as porins. We call them as porins. What do we call them? We call them as porins, right? Pore like structures they are. Are you getting my point? Pore like structures they are. Are you getting my point? Are you getting my point? Done? Done? So basically, right, this outer membrane, when you talk about the outer membrane, some protein channels are present here, some proteins are present here, and what do we call it? We call it as porins. We call it as porins, porins, porins. And through these porins, let's say if water crosses, right, if water movement is there through porins, we use the word aquaporins. So it's not just the water that will move through it, even some ammonium ions can also cross it, right? So basically you know that there are things which cannot cross the membrane directly, right? So for them we have the ion channels. So same way, right, one porins, these proteins are also making some pore like structures in the outer membrane through which the things will move. That is also related to this permeability thing. The outer membrane is more permeable than the inner membrane, okay? And if you read the NCERT properly, in the cell chapter it is given, right, in the cell chapter it is given that when we compare the permeability of the mitochondria and chloroplast, one thing is a fact in both the cases, in both the organelles, that outer membrane is more permeable than inner membrane. When we say inner membrane, right, we say that inner membrane is selectively permeable. It will not allow the movement of everything or anything, right? It is basically selectively permeable. It is as per the needs. It is as per the needs. Right. So when you talk about the mitochondria and the chloroplast, so inner membrane of mitochondria is comparatively, yeah, you can say that the inner membrane of chloroplast, relative to the mitochondria, it is less permeable. Okay? Inner membrane of the chloroplast, as per NCERT cell chapter, right, inner membrane of the chloroplast is relatively less permeable than the mitochondria. That is how we compare. So last time in NEET 2024, right, you know that regarding this a kind of some teachers were saying nahi, this question will give you the bonus marks or something, but actually it was not there. Right? So do read such kind of things and try to relate the things, okay? Right, for your better understanding, if you really want to master your subject, right, bache, then you should know how to relate one with another. You should know it, okay? You should know it. Is that clear? Is that clear? So that is something you need to keep in your mind, that outer membrane is more permeable than inner membrane. It is having the porins, some pore-like structures. The proteins are making it. If water is crossing through that, we are using the word aquaporins. Okay? And along with that, ions can also cross. Just say, I have given you the example of ammonium ions also. Ammonia can also cross through it. Right? And I'll again check it out. And now when we compare the inner membrane of the mitochondria and chloroplast, so we are saying that chloroplast inner membrane is relatively less permeable. Any doubt? Any doubt? So this is the matrix part where the Krebs cycle will occur, right? And this is the inner membrane where your electron transport system is present, where all that proteins, complexes are present. Is that clear? Is that clear? So if pyruvate, yes, if pyruvate will get the oxygen, okay, so this pyruvate has to enter in the Krebs cycle, right? It has to enter in the Krebs cycle, but it cannot do it directly. So in between there is one more reaction that is known as link reaction. That is known as link reaction. That is known as transition reaction. That is known as your gateway reaction. Okay, so if our pyruvate is interested in going for the aerobic respiration, so before Krebs cycle, what will be there? This particular reaction. Matlab, link reaction, you can also call it as transition reaction. You can also call it as gateway reaction. Is that clear? Is that clear? So these are the different names for the same reaction. Okay, these are the different names for the same reaction. Is that clear? Yes. Yes. So the very first step, so if oxygen is available, right? Then pyruvate will go to mitochondrial matrix, and in two steps, in two ways, right? It's further. What is going to happen? It will enter the mitochondrial matrix where its oxidation will be completed in two phases. Okay? Okay? So if oxygen available, pyruvate enter in mitochondrial matrix where its oxidation is completed in two, where its oxidation will be completed in two phases. Okay? So very first phase, जैसे we are calling it as link reaction also. Basically what is happening here? Acetyl coenzyme A forms from pyruvate. Remember this. What's going to happen in this link reaction, bache? What is going to happen in this link reaction? Right, the acetyl coenzyme A will form from the pyruvate. And here in Krebs cycle, what is going to happen? Acetyl coenzyme A enters Krebs cycle, and what will happen? Carbon dioxide and H2O will be released. Carbon dioxide and H2O will be released. Are you getting my point? Are you getting my point? Yes? Yes? So if oxygen is available, your pyruvate will enter the mitochondrial matrix, right, bache? So here in two phases, its oxidation will be completed. First is your link reaction, your transition reaction, or your gateway reaction, in which acetyl coenzyme A will form from the pyruvate, and then acetyl coenzyme A will enter in the Krebs cycle, and CO2 and water will form. Revise it. Revise it. Close your eyes, recall everything. We started the story from the glucose 6-phosphate, right? Fructose 6-phosphate, then fructose 1,6 bisphosphate, then there will be the cleavage, glyceraldehyde 3-phosphate, dihydroxyacetone 3-phosphate. Dihydroxyacetone phosphate will also get converted into the glyceraldehyde 3-phosphate, and then we will have 2-glyceraldehyde 3-phosphate. That will give us what? That will give us the 1,3-bisphosphoglycerate, that will give us 3-phosphoglycerate, 2-phosphoglycerate, then there will be the phosphoenol pyruvate, and finally there will be the pyruvate. Finally there will be the pyruvate, right, bache? So these are the 10 steps of the glycolysis, and you know that in the step 7th and the 10th, substrate level phosphorylation will be there. So when the 1,3-bisphosphoglycerate is getting converted into the 3-phosphoglycerate, then there is a substrate level phosphorylation, 2 ATP will form, and when phosphoenol pyruvate is getting converted into the pyruvate, then again the 2 ATP will form. That's what you need to keep in your mind. That's how you guys can revise the glycolysis. Are you getting my point? That's how you people can revise the glycolysis. That is what you have to discuss. Now, if oxygen is available, it will go for the gateway reaction, then the Krebs cycle, where both the things will occur in the mitochondrial matrix. So your pyruvate is there in the cytoplasm. Firstly, it has to enter in the matrix. We are not talking about that part yet. We are not talking about that part yet. In the higher studies, we discuss it in a little different way. Okay? In higher studies, we discuss it in a little different way. Okay? But tell me, up to this part all clear? Up to this part all clear, or you guys are having any doubt? Yes? Right? So in the mitochondrial matrix, your pyruvate will first oxidize into the acetyl coenzyme A. Okay? Okay? Right, so this link reaction is actually the oxidative decarboxylation. Can you tell me what is the meaning of oxidative decarboxylation? So we are using the word oxidative decarboxylation. What is the meaning of that? What is the meaning of that? You please try to elaborate it. You please try to elaborate it. Yes, bache. So I am saying your pyruvate in the mitochondrial matrix, right? Now this pyruvate is, where is it? It is in the mitochondrial matrix. It is in the mitochondrial matrix. So what is going to happen here? Here this pyruvate will undergo oxidative decarboxylation. So please define this thing. Okay, what is the meaning of oxidative decarboxylation? Anybody in the class? What is the meaning of oxidative decarboxylation? Yes, please answer it to me. I'm waiting for your answers. Quickly tell me. Yes? Tell me what is going to happen here. It is a decarboxylation. Anybody? Very good. Removal of CO2 will be there. But why that oxidative word is there? Why that oxidative word is there? Yes, why that oxidative word is there? That is what I'm asking. So it is very simple, right, bache? It is very simple here. Very simple point here. Let me tell you what is happening. Dekho, oxidative decarboxylation, matlab ultimately carbon dioxide will be removed. So what is happening here? You have two pyruvate. Please remember this reaction, it is much needed. There is an enzyme complex and that is pyruvate, anybody in the class? Dehydrogenase complex. What is the name of the enzyme complex here? What is the name of the enzyme complex here? The enzyme complex here is pyruvate dehydrogenase complex. Pyruvate dehydrogenase complex. Bache, when we are saying this enzyme, matlab pyruvate dehydrogenase complex means this enzyme is made up of many proteins. Many proteins are together. Many different proteins are together making this enzyme. And obviously magnesium ion is again required here. Right? Magnesium ion is again required here. Bache, your pyruvate is a 3-carbon containing compound. As I said, in this reaction, the decarboxylation will occur. Matlab CO2 will release. CO2 will release for sure. Right? Carbon dioxide will release for sure. Right, bache? So carbon dioxide will be released from the pyruvate. We have 2 pyruvate, so 2 CO2 molecule will be released. And how that decarboxylation will occur? Because of the oxidation. Because of the oxidation. From this 2 pyruvate, in this reaction, we will see that 2-acetyl coenzyme A will form. Right? That pyruvate, which is a 3-carbon containing compound, will get converted into the 2-acetyl coenzyme A, right? And that acetyl coenzyme A is a 2-carbon containing compound, and 1 carbon will be lost will go out in the form of CO2. So here from the pyruvate, that C double bond O wala group, that carboxyl group will be removed in the form of this carbon dioxide. Right, and moreover, here you will see that NAD will also be oxidized, and you are going to get 2 NADH2 here. Right, we also get 2 NADH2 here. Are you getting my point? We are also getting 2 NADH2 here. Right, bache? So because it is acetyl coenzyme A, so definitely we have to add coenzyme also, so that we can get this coenzyme A. So this is the link reaction. And why this reaction is important? Because your pyruvate cannot directly enter in the Krebs cycle. This pyruvate has to get converted into the acetyl coenzyme A, then only it can enter in the Krebs cycle. So 2 pyruvate, because of this pyruvate dehydrogenase complex, right, and obviously magnesium ion is required, it will form 2-acetyl coenzyme A. It's a 2-carbon containing compound. It's a 2-carbon containing compound, and the CO2 is released. 2 NADH2 will also form. 2 carbon dioxide will also release. This is the link or the transition reaction. The same is given in the NCERT as well, and this is the reaction. This is the reaction in the NCERT. Firstly, let me explain this, then we will go ahead. So here 1 pyruvic acid, you can also write pyruvate, it's fine. Coenzyme A, 1 this oxidized NAD. Pyruvate dehydrogenase, magnesium. Then acetyl coenzyme A, carbon dioxide, NADH2. Again, I am repeating this, right? It's a 3 carbon containing compound. This one is oxidized. This is a 2 carbon containing compound. This one is reduced. Okay? And now we know that we have 2 pyruvates from the glycolysis. So that's why I'm writing 2 pyruvic acid, 2, 2, 2, 2 and 2. So can I say that? Can I say that? Yes, you tell me. You tell me guys. Can I say that from one glucose, because we are getting 2 pyruvate, right? So that 2 pyruvate, they'll get converted into acetyl coenzyme A. Acetyl coenzyme A, and that acetyl coenzyme A will enter in Krebs cycle. Yes, can we say so? Can we say so? Can we say so? Can we say so? So can I say that कि one glucose molecule for its complete oxidation, it is undergoing 2 Krebs cycle, right? Can I say so? One glucose molecule, it is undergoing 2 Krebs cycle. Yes, it is undergoing 2 Krebs cycle. Can I say so? One glucose molecule is undergoing 2 Krebs cycle. Can I say so? You tell me. Because we have 2 pyruvate, 2 acetyl coenzyme A, 2 times this cycle will occur. 2 times this cycle will occur. And here, in this step also, we got, right here, in this step also, we got this. Here in this step also, we got total. Am I clear? Am I clear here? If there is any doubt, do let me know. If there is any doubt, do let me know. Done? Done? Okay. So this part is clear. This part is clear. Okay. Let's cover it from the NCERT and then let's start the Krebs cycle. And trust me, now you are going to love the topics. They are amazing. They are very simple. For aerobic respiration to take place within the mitochondria, final product of glycolysis, that is pyruvate, we have to transport it from the cytoplasm into the mitochondria. Okay, so there are the crucial event in aerobic respiration are first the complete oxidation of pyruvate by the stepwise removal of all the hydrogen atoms, right? We have to remove the hydrogen. Who's going to accept that hydrogen? Your NAD positive, which is oxidized, right? So leaving 3 molecules of carbon dioxide. They are saying when there is the complete oxidation of pyruvate by the stepwise removal of the hydrogen atom, leaving 3 molecules of carbon dioxide. Never ever get confused here, right? See, because they are talking about only 1 pyruvate. Okay? So 1 pyruvate, when it is giving you 1 acetyl coenzyme A, then 1 CO2 will be removed. Then one CO2 will be removed. Remaining 2 CO2 will be removed during Krebs cycle. Remaining 2 CO2 will be removed during Krebs cycle. That is why they have written 3 molecules of CO2. Right? So they are saying when there is a complete oxidation of the pyruvate by the stepwise removal of H positive. So who's going to accept that H positive? NADH, and we'll see FADH2, right? And 3 molecules of CO2 will be released from this pyruvate. So 1 molecule will release here during link reaction. Remaining 2 CO2 will release during Krebs cycle. Doubt? Doubt? Next, the passing on of the electrons removed as the part of hydrogen atoms to molecular oxygen with simultaneous synthesis of oxygen, ATP. So ultimately that electrons or the hydrogen, ultimately the passing on of the electron removed as the part of hydrogen atoms to molecular. Ultimately your terminal acceptor, your final acceptor is the oxygen. That is going to accept it. In the electron transport system, here they are talking about the electron transport system where the electrons will transport, the energy will be generated, will move the protons, and finally, right bache, there is the oxidative phosphorylation that will result in the synthesis of ATP. So you will see, we will even compare it with the photosynthesis also. Right? You will get to know about it in a better way. So what is interesting to note is that the first process take place in the matrix. Right? What is interesting to note here, that the first process, that is your link reaction, that is your Krebs cycle, it will be there in the matrix, right? And the second process is there in the inner membrane. So second process here means you are talking about the electron transport chain. We are talking about the electron transport chain. Okay? So pyruvate, which is formed by glycolytic catabolism, enters into mitochondrial matrix. It will undergo oxidative decarboxylation by a complex set of reaction catalyzed by pyruvate dehydrogenase. Any doubt here? Any doubt here? Right, so for this reaction you need the presence of several coenzyme NAD and coenzyme A. Fine, so this is your mitochondria, the structure here. Right, so ATP synthase, oxysome, F0, F1 is same. Okay, ATP synthase, oxysome, F0, F1 particle is all same. Okay, it's ATP synthase. Try to correlate with the chloroplast where it was CF0, CF1. Okay, so this is the wonderful diagram. This is that wonderful diagram. I'll use this diagram not now, when I will explain you the electron transport chain. So these are that enfoldings, these are that cristae, right? Can you see that how well the things are shown here in this diagram? Right? So this is showing the inner membrane, this is showing the outer membrane. So here in this outer membrane, the porins are there by which the things will move. Look at this, look at this. This is your pyruvate, this is your pyruvate. This pyruvate has to enter in the mitochondria, right? Through this it is entering into the mitochondria and finally here in the matrix, right? Finally here in the matrix. This reaction, pyruvate dehydrogenase complex, it is forming the acetyl coenzyme A. Right, further here in this reaction they are showing how different other compounds like fatty acid, right, how are they forming this acetyl coenzyme. So once we'll complete it now, I'll discuss it. I'll discuss everything from this particular diagram, and trust me, you are going to love it. Right, you are going to love it. Here they are showing that Krebs cycle, the complete Krebs cycle. Here this enzyme, this is showing the complexes of inner membrane where electron transport system will there, and this is the ATP synthase which will help in the ATP formation. Okay, so that's what we have to discuss. Are you ready for that? Are you ready for that? Okay, so during this process, 2 molecules of NADH are produced from metabolism of 2 molecules of pyruvic acid. So in the reaction they are showing 1 pyruvate, that's why 1 NADH, but in the text they have mentioned it 2 molecules of pyruvate, right? So 2 NADH. Is that clear? So it is the acetyl coenzyme A that will enter the cyclic pathway, that is your Krebs cycle, right? So remember, there are different names of this Krebs cycle. This Krebs cycle is also known as tricarboxylic acid cycle. Will tell you the reason as well. Tricarboxylic acid cycle, and we also call it as citric acid cycle. It is also known as citric acid cycle, right? So Krebs cycle, tricarboxylic cycle, या फिर you can call it as TCA, tricarboxylic acid cycle, या TCA cycle, or the citric acid cycle is all same, right? It is all same. These are the different names here. Okay, बच्चे? Okay, बच्चे? Okay बच्चे, so tricarboxylic acid cycle, Krebs cycle, tricarboxylic acid cycle, TCA cycle, Krebs cycle, and citric acid cycle is all same, right? And the citric acid cycle is all same. Is that clear? So Mr. Hans Krebs was the one who first elucidated it. So up to this part all clear? Up to this part all clear? This thing should be clear, that what are you getting from the glycolysis, what is there in the link reaction, and now we will talk about the Krebs cycle. So if it is clear, I want to see some fire in the chat section. I want to see some fire in the chat section. Quick, I want to see the fire in the chat section. Done? Sure? Very good, very good, very good. So for the Krebs cycle, see, first of all, when you are saying Krebs cycle, here in this cycle, this is a cyclic process. Remember, this is a cyclic process. What is it? Cycle. It is a cyclic process. So we know that when we talk about a cyclic process, जैसे A to B, B to C, C to D, D back to A, right? So here in the Krebs cycle also you will see that the substrate entrant, right, the one that will enter here in the Krebs cycle, it is acetyl coenzyme A, a 2 carbon containing compound. It is acetyl coenzyme A, a 2 carbon containing compound. And who's going to accept this? The acceptor here is your 4 carbon containing compound, that is oxaloacetate or oxaloacetic acid, right? Right. So here in the Krebs cycle, the substrate entrant is your acetyl coenzyme A, and who's the acceptor, or you can say that the first acceptor is the oxaloacetate, and obviously the first product, we also always talk about the stable one. So the first product is the 6 carbon containing compound citric acid cycle. Right, citric acid cycle, that's a 6 carbon containing compound. So now there is no confusion here. ठीक है बच्चे? Now there is no confusion here. Right, now there is no confusion here, isn't it? Isn't it? What is there? There is no confusion here. So देखो, substrate entrant is a 2 carbon containing compound. Substrate entrant is a 2 carbon containing compound. First acceptor is the oxaloacetate. Right बच्चे, the first product is 6 carbon containing compound. मतलब 2 carbon containing compound is entering, 4 carbon containing compound is accepting, is 4 plus 2, 6 carbon. 6 carbon, there is no doubt here. And it is a cyclic process. So whosoever is the acceptor, we have to regenerate it to continue this process. Okay, so this Krebs cycle is also a 10-step process, but बच्चे, we mainly talk about the 7 steps, right? We mainly talk about the 7 steps. It is also a 10-step process. But mainly we will see that 7 and 8 steps here. Okay, and this particular process is amphibolic. Like if I have to focus on this Krebs cycle, what kind of process it is? It is amphibolic. So what is the meaning of being a amphibolic process? Amphi means dual. Right? So both the things will be there. The catabolism, the anabolism. Both the things are happening in this particular process. So substrate entrant, acceptor, product. This is what we have to discuss. Now, the trick here is, right, come in. I know the trick is not that good, but theek hai yaar, that's how I remember this. So come in and kick. Scooter, then fastly move on. ठीक Okay, what's the trick? Please repeat it in the chat section, and you can just search it on YouTube. You can just add my name and you can see the short video as well. Right? You can even see the short video as well. Okay, bache? So what is the trick? Come in and kick scooty scooter, fastly move on. Come in and kick scooty scooter, fastly move on. Come in and kick scooty scooter, fastly move on. So quickly, come in and kick scooty scooter, fastly move on. Right? Repeat it in the chat section. Done. Do it quickly, guys. Please do it quickly. Dekho, if you will focus now, as I said, this chapter is not difficult, right? We need extra like 1.5 hours without break to finish, right? We don't have too much here. Chali, do it. Come in and kick scooty scooter, fastly move on. So come in. C stands for citrate. C stands for citrate. I stands for isocitrate. They are isomers also. And kick from this A and K. Alpha ketoglutarate. It's alpha ketoglutarate. Okay? Scooty. You have succinyl coenzyme A. Succinate. Okay? Then F stands for fumarate. Then M stands for malate, and then O stands for oxaloacetate. O stands for oxaloacetate, right? O stands for oxaloacetate. So it's come in and kick scooty scooter, fastly move on. Come in and kick scooty scooter, fastly move on. Come in and kick scooty scooter, fastly move on. So let's repeat it again. So C stands for citrate. C stands for citrate. So this citrate is a 6-carbon containing compound. This is the isocitrate. It is the isomer here. Obviously, it will also be the 6-carbon containing compound. Right? Then you have and kick. From and kick. So dekho na bache, you have to remember this. Right? You have to remember this. And kick means we are picking up A and K. Alpha ketoglutarate. It's a 5-carbon containing compound. And then it is the succinyl coenzyme A. And then it is the succinyl coenzyme A, which is a 4-carbon containing compound. Your succinate, right? Your succinate, again 4-carbon containing compound. Fumarate, again 4-carbon containing compound. Okay? Your malate, again a 4-carbon containing compound. And if you guys know the photosynthesis, you already know that oxaloacetate, this malate, they all are 4-carbon containing compounds. Right? They are like this. They are like this. So is it easy or difficult? Is it easy or difficult? It is easy. It is easy. Okay? So we already know that first compound is the citrate. That's why it is the citrate cycle or citric acid cycle. It is 6-carbon. So we have there is no need to get worried. We know that first acceptor is oxaloacetate, 4-carbon containing compound. So you just need to remember that it is the alpha ketoglutarate, which is a 5-carbon containing compound. Rest all are 4 or 6. So say in the 6-carbon containing compound, you are not going to get any confusion. Why? Because first product is a 6-carbon containing compound, citrate. Citric acid cycle, and isocitrate is it's what? It is its isomer, so 6-carbon containing compound, right? So you just need to remember alpha ketoglutarate is 5-carbon, and it will be clear in your mind ki 6-carbon is this, remaining all will be 4-carbon containing compound. 4-carbon containing compound. Chalo, now I'll ask you one question. If from isocitrate, if from isocitrate, if alpha ketoglutarate is forming and 5-carbon, 6-carbon is getting converted to 5-carbon, don't you think the decarboxylation occur here? The CO2 will release here. Don't you think so? The decarboxylation will occur here. The CO2 will release, definitely. Now from this 5-carbon containing compound, you will see 4-carbon. So there will be again decarboxylation. Means again the carbon dioxide will be released. So that is also a PYQ, that in the Krebs cycle, how many steps of decarboxylation are there? 2. How many steps of decarboxylation are there? 2. Understood? 2. Right? So come in and kick scooty scooter, fastly move on. So you already know it is the citrate. It is the citrate that is the first stable compound. So citrate, 6-carbon containing compound. Isocitrate, 6-carbon containing compound. Alpha ketoglutarate, 5 carbon containing compound. Succinyl coenzyme A, 4 carbon containing compound. Succinate, 4 carbon containing compound. Fumarate, 4 carbon containing compound. Malate, 4 carbon containing compound. Then finally oxaloacetate, 4 carbon containing compound. This is that complete cyclic process. This is that complete cyclic process. So tell me, is there any difficulty here in the Krebs cycle? It is amphibolic. It is amphibolic. Right? Right? Yes, bache. Right? And now you know that the acetyl coenzyme A, a 2 carbon containing compound, is the substrate entrant here. Okay? So guys, the first enzyme used here is your citrate synthase. Is it synthase or is it synthetase? Anybody in the class? Is it synthase or is it synthetase? Anybody in the class? Anybody in the class? Because it is something very important. Yes, it is something very important. It is the first step that we are talking. Is it citrate synthase? This reaction, who's going to catalyze? Citrate synthase or synthetase? It is synthase. It is synthase. So here in the synthase, the ATP is not used. When the enzyme is synthetase, that will be the one. So this is the ATP, sorry, this is the synthase. So synthase and synthetase, they are different. Okay, synthase and synthetase, these two enzymes are different. Is that clear? Is that clear? Is that clear? So synthases, they are the one that do not use the ATP for the reaction, but your synthetase, right? Right, they use this ATP and all. Okay? Right, so citrate synthase, this the synthase category of the enzyme, it doesn't use the energy from that ATP, GTP and all. But synthase, they do not use it, but synthetase, they use it. So synthase and synthetase, these are two different terms. If you will write citrate synthetase here instead of synthase, you are making mistake. You are making mistake. So what should be there? What should be here? Here it should be synthase. Citrate synthase. What should be there? Here it should be synthase. Right? So synthase will not use the ATPs, GTP, etc., but synthetase, they use it. Okay? So this is the Krebs cycle, right? This is the Krebs cycle, a cyclic process, a 10-step process. But here you will be like, ma'am, 1, 2, 3, 4, 5, 6, 7. We are not discussing about the 10. Actually, here in between these two, cis-aconitate form, and here in these two, oxalosuccinate form. So we do not talk about that. Okay? Even in NCERT, it is not given properly. So we do not talk about that. Okay? So now, if it is clear to you, then let's move to this. This particular cycle. Now, we are talking about the citrate. We will first talk about the citrate here. As I said, it's a 6 carbon containing compound. As I said, it is a 6 carbon containing compound. But why do we say that Krebs cycle is also known as tricarboxylic acid cycle? Why do we say that Krebs cycle is known as tricarboxylic acid cycle? Tri means 3 carboxylic acid group, COH, COOH, COOH. And because it's a 6 carbon containing compound, look at the structure here. So you guys will see hydrogen, hydrogen, OH, hydrogen and hydrogen. That's it. This is the structure here. One second, I made one mistake here. Now it's fine. Just look at this. It is your citric acid. What is it? Citric acid. Why am I saying it is citric acid? देखो, citric acid is a 6 carbon containing compound, and the citric acid is actually having 3 carboxylic acids, COH, COH, COOH, COH, COH, COH. That's why it is the tri, means 3 carboxylic acid cycle. The first product here is this, right? It is having 3 carboxylic acid plus. Why am I saying it is not citrate and citric acid? Because see, it is not in the ionic form. So CO negative, CO negative, CO negative. Now it is the citrate, right? Now it is the citrate. Now it is the citrate. So that is why we call it as citric acid cycle. That is why we call it as tricarboxylic acid cycle. Okay? That is why we call it as citric acid cycle. That is why we call it as tricarboxylic acid cycle. Is that clear? Is that clear? Right? Is that clear? Sure? Sure. So oxaloacetate, 4 carbon containing compound. Oxaloacetate, 4 carbon containing compound. So I'm not saying that you have to remember the structures. If you know it, good. It's your choice. So this is your acetyl coenzyme A, your citrate. So citrate will form isocitrate. Isocitrate will form alpha ketoglutarate. Succinyl coenzyme A, succinate, fumarate, malate, and oxaloacetate. Firstly, remember this. Remember this cycle. Then I will tell you where will you see the substrate level phosphorylation, where will you see the decarboxylation, and where that NAD and FADH2 will form. I'll tell you about it. So this thing clear? Why do we call it as tricarboxylic acid cycle? That why do we call it as tricarboxylic acid cycle and the citric acid cycle? I hope it is clear. I hope it is clear. Done? Done? So the things that are there in front of you, right? On the basis of that, let's learn something. Let's learn some things, right? On the basis of that. Your isocitrate is giving you alpha ketoglutarate. Alpha ketoglutarate. That's a 5 carbon containing compound. So obviously the decarboxylation is taking place. The decarboxylation is taking place, and here, because here you will see NADH2 will also form. Now from alpha ketoglutarate, you know that the succinyl coenzyme A is forming, again 4 carbon containing compound, again decarboxylation, and again bache, again here also you will see that NADH2 will form. So remember that NCERT line where it is written ki from the complete oxidation of pyruvate will release 3 carbon dioxide molecules. So 1 in the link reaction, 2 will be here. 2 will be here. Is that clear? Is that clear? Okay? Okay. So you should know this. Now when we convert the succinyl coenzyme A into the succinate, so this is the substrate level phosphorylation. This is the substrate level phosphorylation. So it is also a previous year question, bache. It is also a previous year question that in Krebs cycle, how many steps of substrate level phosphorylation are there? Only 1 step of substrate level phosphorylation is there. Only 1 step of substrate level phosphorylation is there. What is there? Only 1 step of substrate level phosphorylation is there. Okay? Okay. So I told you what is going to happen. Your succinyl coenzyme A. Okay, I'm writing it here so that you can just remember this. We will convert it into succinate, right? 4 carbon containing compound, 4 carbon containing compound. And which enzyme is there? Do remember this, because individual reactions, right? They can ask the individual reaction here, right? They can ask it, right? They can ask about the individual reaction here. So when we are converting succinyl coenzyme A to succinate, it is a kind of 5th step it is, right, bache? So here the enzyme used will be succinate. Yes, because it is the substrate level phosphorylation. So I believe you guys can use your brain here. So it is the succinate, but it is the succinate, bache, thiokinase. The enzyme here is succinate thiokinase. And obviously succinyl coenzyme A is giving succinate. So coenzyme A will also come out. So actually here what is happening, let's go for, so substrate level phosphorylation, you know where the substrate is providing the energy to add the phosphate. So GDP, here we talk about the GDP, that will be converted into the GDP. GDP means guanosine diphosphate. Guanosine diphosphate. So that guanosine diphosphate will get converted into the guanosine triphosphate. It will get converted into the guanosine triphosphate. And bache, actually this reaction, immediately this GTP, what will it do? Again, it will just release its phosphate, it will form AD. Basically here, right, that is how we write it. One second. This, I'm talking about this step. I am talking about this step. See this. In the NCERT, it is not given properly. See this. So when succinyl coenzyme A is forming the succinate, GDP is giving us the GTP. So here one explanation is there that we should know, because question, they can ask the question from this part. So it is saying that during the conversion, right bache? It is saying that Arey? Okay. Yes. So it is saying that during the conversion of succinyl coenzyme A to succinate, a molecule of GTP synthesized. Now I am explaining the NCERT, kindly pay attention. Right, what is happening here? Ki during the conversion of succinyl coenzyme A to succinate, during the conversion of succinyl coenzyme A to succinate, a GTP is synthesized. This is a substrate level phosphorylation, very famous question. In a coupled reaction, this GTP will again convert it to GDP with the simultaneous synthesis of ATP. Matlab, this GTP will give us ATP. How? Because when this GDP will form GTP, simultaneously, immediately it will get converted to GDP. And when it will get converted to GDP, matlab it has released its phosphate, that will be picked up by the ADP and it will form ATP. That is what, right, that is what they are trying to explain here. Okay, that is what they are trying to explain here. So what are they saying? What are they saying? The substrate level phosphorylation is there. The enzyme is succinate thiokinase. And we can also call this enzyme, the another name of this enzyme is, succinate coenzyme A synthetase. I believe that's easy for you, but it is synthetase, not synthase. Okay? So you can call it succinate thiokinase, or we can also call it succinate coenzyme A synthetase. Is that clear? We can also call it succinate coenzyme A synthetase. So ADP and GTP, matlab they are energetically, on the basis of energy, they are equivalent. Right? Energetically, they are equivalent. But here, what you need to remember as per NCERT, that this GDP will immediately get converted to GDP. It will release its phosphate. ADP will pick it up and ATP will form. So that's what you have to remember. Okay? Okay? Yes. Done? Okay? Done, bache? So remember one thing. In animal cell, actually, there are two isoenzymes here. I'll add this thing here. Like when you talk about the animal cells, na, in the case of animals, right? When you talk about this particular step, in the case of animals, when you talk about this particular step, so animal cells, they have two isoenzyme of succinyl coenzyme A synthetase, right, bache? They are having two isozymes of succinyl coenzyme A synthetase, and one is specific for GDP, one is for GDP and one is for ADP. ठीक है? I know you will be like, madam, not given in NCERT, why are you telling this? Because I'm explaining something very simple here. कि animal cell में there are 2 isozymes of succinyl coenzyme synthetase, one specific for GDP and other is for ATP. You can write the word specific also. One specific for GDP and another is for ADP. Right, 2 isozymes are there. Right, 2 isozymes are there. So GTP that will be formed here can donate. मतलब the GTP, though energetically both are energetically GTP and ATP, they are equivalent, but the GTP that form here, right, जैसे here I am saying the GTP that form here, it can transfer its terminal phosphoryl group to the ADP. जैसे the same thing NCERT is explaining कि when the GTP will form, it will immediately get converted to GDP. So who's going to accept that phosphate? The ADP. It will form ATP. So that is the story of the animal cell. That is the story of the animal cell, right? So because they are talking about the respiration in plant, they should not pick that up. They should not pick this point up. So in plants, even in bacteria, in plants, even in some bacteria and in some animal tissue, in plant, in bacteria and some animal tissues, what is the story in their case, bache? What is the story? An ATP molecules forms directly by substrate level phosphorylation, right? An ATP molecule form directly by substrate level phosphorylation. Understood? Understood? Sure? Hello? Sure? So let's revise it once again. The trick is come in and kick scooty scooter, fastly move on. The trick is come in and kick scooty scooter, fastly move on. So that is saying citrate, isocitrate, alpha ketoglutarate, succinyl coenzyme, succinate, fumarate, malate and oxaloacetate. That's how you have to write. It's very simple. If isocitrate is 6 carbon containing compound and it is giving us alpha ketoglutarate, that is a 5 carbon containing compound. Matlab here the decarboxylation is there. And it is the step where you people will see that NADH2 will form. Okay? Then alpha ketoglutarate is forming 4 carbon containing compound. Then again decarboxylation is there. Here also you will see that NAD will give us NADH2. Now when we are converting succinate into succinyl coenzyme into succinate, there is a substrate level phosphorylation. Here we will get 1 ATP. Sorry, here we will get 1 ATP. I hope it is clear. I hope it is clear. Now we are converting the succinate into the fumarate. Succinate into the fumarate. F. From this F, you have to remember that here you have FAD positive, oxidized FAD, that will give FADH2. That will give FADH2. So what is this FAD? Flavin adenine dinucleotide. Right? Flavin adenine dinucleotide. Right? Okay. And now after that, when malate is getting converted into oxaloacetate, here you will again see that 1 NAD is oxidized to NADH. Is that clear? That's what we have to remember. That's what we have to remember. Come to this particular diagram. Come to this particular diagram. So this part clear? Right, I'm not talking about the enzymes at this point of time. The reason is I want you people to understand that first. Right. So oxaloacetate is the acceptor. It will accept acetyl coenzyme. It is giving us citrate. That citrate will get converted to isocitrate. Dekho, enzymes are also simple. Oxaloacetate is forming citrate. Citrate synthase. Citrate synthase is the enzyme there. Citrate is forming isocitrate. Aconitase is the enzyme here. In between, there is a kind of transition reaction. We will talk about it because it is a PYQ. Now, isocitrate will get converted to alpha ketoglutarate. So, name of the enzyme is also same because here NADH forms. So, you can say isocitrate dehydrogenase. Alpha ketoglutarate is giving us succinyl coenzyme. Again, NADH will form here. And the decarboxylation is here. So alpha ketoglutarate dehydrogenase. And right now I told you succinyl coenzyme will form succinate. It is the succinyl coenzyme succinate, the synthetase, or we can also call it as synthase. Sorry, we can also call it as succinate thiokinase. Okay? succinate thiokinase. Okay, so up to this part all clear, up to this part all clear, up to this part all clear, guys. If it is a yes, I want to see some fire emojis in the chat section, then I will move to the next step. Quick. Quick. Done. So then your succinate will, now see, succinate is giving you the fumarate. It is also interesting. Succinate is giving you the fumarate. This reaction is also interesting. So whatsoever is very important now, I'm writing it separately. So I want you people not to miss that. So succinate is going to form fumarate. Both are 4-carbon containing compound. So definitely there is no decarboxylation. But here you will see that there is an enzyme, that is succinate dehydrogenase. Who's catalyzing this reaction? Succinate dehydrogenase. Who's catalyzing this reaction? Succinate dehydrogenase, right? Succinate dehydrogenase. So what is special about this enzyme here? What is special about this enzyme here? See, all the enzymes of Krebs cycle are in mitochondrial matrix. All the enzymes of Krebs cycle, they are in mitochondrial matrix, right? Where are they? They are in mitochondrial matrix except, right? Except succinate dehydrogenase, right? Except succinate dehydrogenase, right? So this enzyme is present in the inner membrane of mitochondria. This enzyme is present in the inner membrane. Yes, in the inner membrane of mitochondria, right? And it is actually the complex 2 of that. It is actually making the complex 2 of that electron transport chain. Is in electron transport chain we talk about the complexes. So that second complex is the succinate dehydrogenase, right? It is actually the complex number 2 of electron transport chain. So this particular enzyme, we say that it links Krebs cycle to electron transport chain or electron transport system. Isn't it interesting? So succinate is giving us the fumarate. Succinate is giving us the fumarate. So FAD is here. FAD is forming, and this FAD, oxidized FAD is accepting the electron. It is forming the FADH2 and it is flavin adenine nucleotide. Okay? So this reaction is also important. This reaction is also important. Is that clear? So there is also a special point about this reaction. So whatsoever is important and from where they ask the questions, I'm writing it here. Okay? I'm writing it here. So you should know about it. You should know about it. Is that clear? Is that clear? So that fumarate is giving you malate, and that malate is giving you oxaloacetate, right? That is giving you oxaloacetate. So here just remember this, that when we are regenerating oxaloacetate, one more NADH will form. That's it, that's all. That's it, that's all. So this is about the Krebs cycle, right? One more cycle. See, I'm telling you that there is one, just see, this citrate is forming isocitrate, but in between there is the, right, it is forming isocitrate, but in between there is the cis-aconitate. So this is a PYQ, students, that's why I'm explaining it. Your citrate, before converting into the isocitrate, it is having right? It is having an intermediate structure, yet transition, it is having a structure in between, that is your cis aconitate, and then it forms the isocitrate. So it is a previous year question. It is a previous year question, you people can see this. Okay, it is a previous year question. So this step, both the steps are catalyzed by aconitase. Okay, both these steps are catalyzed by aconitase. So it is a previous year question. Do solve it. You will get it. So citrate will give cis aconitate, and cis aconitate will give isocitrate. So aconitase and aconitase is the enzyme that is catalyzing it. So here you can see your isocitrate is giving you alpha ketoglutarate. So this is also oxidative decarboxylation. Okay, so one oxidative decarboxylation is link reaction. Two steps are here. So I will share the notes in the Telegram group. You will get these amazing pictures, and obviously these kind of pictures, they help in knowing the concept in a better way. Okay, right? So here OH group oxidized to carbonyl, which is turn facilitates decarboxylation by stabilizing carbanion formed on adjacent carbon. So obviously in chemistry form, no need to remember it. So ultimately that's what you have to remember, right? Okay. So alpha ketoglutarate to succinyl coenzyme A, and here also in between there is one step, that is oxalosuccinic acid form. So we are not talking about it, right? Not even a PYQ, that's why. Okay, in between here also you say, right, before alpha ketoglutarate oxalosuccinic acid it forms. So we don't talk about it. That's why we say overall we have how many steps? We have total 10 steps are there. Here also if you will see, they are showing the 8 steps. So 9th and the 10th, I told you about it. Okay, ji, I told you about it, right? So and then this succinyl coenzyme A, succinate, substrate level phosphorylation, enzyme is succinyl coenzyme A synthetase. Then succinate to fumarate, succinate dehydrogenase, right? It is present in the electron transport system. Then your fumarate will form malate. Here you have the fumarase, and then malate will form oxaloacetate. It is the malate dehydrogenase. So here also the NADH will form. Here also the NADH and NADH will form. So yes, this is what you have to remember. So I'm expecting that by tomorrow, because tomorrow also there is no class, you can use that time for the revision, and do not forget about that 24th October event. So you have to come to this channel and you will get the link where you can see that event, okay? Right? So this part is clear, right? This part is clear, right? So this is the Krebs cycle. So now I'm going to write something. The glucose is giving us 2 pyruvate, and I am getting 2 NADH2 here and 2 ATP here. Any doubt? Now 2 pyruvate is giving 2 acetyl coenzyme here, right? And here also we are getting 2 NADH2 and 2 carbon dioxide molecules are released. Can I say so? Now from this acetyl coenzyme A, because it is entering in the Krebs cycle. So Krebs cycle will result in the complete oxidation of glucose into carbon dioxide and water. We know that. So ultimately what is happening, right? What is happening here? In Krebs cycle, ultimately what is happening? CO2 and water is coming out now. So we know that how many CO2? Ye dekho. Because we have 2 acetyl coenzyme A. So 2 times that Krebs cycle will be there. So that's why total 4 carbon dioxide molecules will release, right? Plus total 6 NADH2 will form, 2 FADH2 will form, 2 ATP will form. Tell me if there is any doubt here. This thing should be clear to you. Ye dekho. Again I'm repeating. You have 2 acetyl coenzyme A, right? Let's take it 1. So 1 acetyl coenzyme A, when it is completely oxidized into CO2 and water, what is happening? Actually, basically, 3 molecules of CO2 will release. So, sorry, 2 molecules of CO2 will release. है ना? And 3 NADH2 formed and 1 FADH2, 1 ATP. Now tell me, that's what we have seen in the Krebs cycle. That is what we have seen in the Krebs cycle. So Krebs cycle is telling us about the complete oxidation of that pyruvate. So ultimately we know that from link reaction we will get this 2 carbon containing compound acetyl coenzyme A. And here in this step, right, we have to take out its 2 carbon. We have to completely oxidize it. That's why 2 CO2 are evolved here. Right, and 3 NADH2 formed, 1 FADH2, 1 ATP. But you know that from one glucose, from one glucose we got 2 pyruvate. From 2 pyruvate we got 2 acetyl coenzyme A. That is why we just double it. That is why I'm saying here that we have 6 NADH2, 2 FADH2 and 2 ATP. Any doubt? And 2 ATP. Any doubt? Any doubt? Any doubt? Again, let's start it from here. 1 glucose is giving us 2 pyruvate, and here this is what we got in glycolysis. When there is the transition reaction, this is what we got, right? And when there is the Krebs cycle, that's what we have. Now tell me any doubt. Now tell me any doubt. Any doubt here? If there is any doubt related to it, please feel free to ask. Please feel free to ask. In the old NCERT, they used to say that 1 NADH2 gives us 3 ATP, 1 FADH2 gives us 2 ATP. So answer used to be different. So now this time if you will get that options, accordingly you have to answer. But concept actually is कि in electron transport chain from 1 NADH2 we can get 2.5 ATP, and from 1 FADH2 we will get 1.5 ATP, right? We will get 1.5 ATP. It is like that. It is like that, right? So ultimately, dekho, we have to make the ATP. So these NADH, these FADH, even they have to transfer their electrons, right? And terminal electron acceptor is your oxygen. That is your oxygen, and we need that energy so that we can make the ATP. So even in new NCERT, things are given clearly, right? But the point is, but the problem in the case of new NCERT is कि in the diagram the correction is required. I'll come to this when we will discuss the ETC, right? When we will discuss the electron transport chain, I'll come to that part also. I will come to that part also. Okay, so 1 NADH2 is giving us 2.5 ATP, 1 FADH is giving us 1.5 ATP. That's what you have to, that's what you need to know. So ultimately 6 NADH here, 2 here, matlab 8 plus 2 this. We have to send them to the electron transport chain. And that electron transport chain is in the inner membrane. So next topic that we need to discuss is electron transport chain या electron transport system and oxidative phosphorylation. What's the word? Oxidative phosphorylation. So word is, so see, you know about the, yes, you people know about the photophosphorylation. You know about the photophosphorylation. Yes, we know about the photophosphorylation that we have discussed in the photosynthesis. We know about the photophosphorylation that we have discussed in the photosynthesis, where in the presence of light, the ATP production will be there. Where in the presence of light, for inorganic phosphate, the phosphate will get added to the ADP and we will get the ATP. Now this is the electron transport. And now today we learned about the substrate level phosphorylation. That substrate will provide the energy and we will get the ATP. Here it is oxidative phosphorylation. So energy will come from the oxidation. energy will come from the oxidation and ultimately the ATP will form. Okay, so next topic is electron transport system, in which complex number 1, complex number 2, complex number 3, complex number 4, they are involved. Okay, and then there is oxidative phosphorylation, ultimately formation of ATP, in which your complex 5 is there. Okay, so that is what we need to discuss next. And yes, I will give you a short break. I'll give you a short break, right? But one thing that I would like to mention here is, chalo theek hai, we'll do it later, right? So let's meet after a break. So after a break, in the right, we'll finish this part and then we will move to that RQLU amphibolic thing. That's it, right? So after the break, we need 1.5 hours to finish it maximum. Okay, I know the topic is difficult, so that's why I'm giving you the break here. So let's meet at 10:50. Okay, let's meet at 10:50. Okay. Hi, hello. Hannah, today though you are getting very long breaks, isn't it? Hannah, today you are getting very long breaks, isn't it? So actually, students, even I'm having headache. Okay, but was not interested in cancelling your session because from last 2 days, yesterday, Yava sir has cancelled. Yava sir had cancelled the session. Then Wasim sir, and so I was like, कि चलो यार छोड़ो, कोई बात नहीं, we'll manage, है ना? So I should not end it at least. I should not cancel it at least. Okay, chalo. So now let's discuss about it. So as I said that now from the Krebs cycle, what we have learned that the glucose is completely oxidized, the CO2 and water has formed. We got the ATP also, but NADH2 and the FADH2. Now we have to, from that NADH2 and FADH2, we have to make the ATP, and for that we need the electron transport system and oxidative phosphorylation, right? So here we are going to talk about we are going to discuss about some complexes. Okay, we are going to discuss about some complexes here. I have seen that students, they consider this topic very difficult. For them, electron transport chain is a problem, ma'am. We are not able to understand it. So honestly speaking, I am not going to enter in the details of that, but whatever is required, right, to answer the questions that can come in the NEET examination, according to that I'm going to teach you this topic. Okay, according to that I'm going to teach you this topic. For an example, they can ask you that how electrons will move from NADH2, from they'll pass to which complex, what should be the route there. I'll discuss that, right? We'll talk about the position of the complexes. We'll discuss about the electron carriers, about their movement as well. Okay, bache? Okay, bache? And we will also discuss that how many ATP will produce from NADH2 and FADH2. Okay, bache? Okay, so see, in the photosynthesis also, this is a portion that we have covered, right? In the photosynthesis also, if you remember, right? If you remember the Z scheme, right? I'll relate it a bit with the photosynthesis part also. If you remember, this is the chloroplast. Here we have the thylakoid, right, bache? This is the granum, and you know that these sac-like structures are thylakoid. So we know that, right? We know that. Now what is the point that we have? What is the point that we have? Just say here, this is the stroma. What's that? It is the stroma, right? Here we have the circular DNA. What do we have here? Here we have the circular DNA, and we have the 70S ribosome. What do we have here? 70S ribosome. Now, what's the point here? Here you have the thylakoid lumen. Here, what do you have? Here you have the thylakoid lumen. ठीक है जी, thylakoid and thylakoid lumen is there. Now, if you remember, right, the thylakoid membrane, this thylakoid membrane is having the protein complexes. This thylakoid membrane is having the protein complexes. What do we have in the thylakoid membrane? If you remember, the protein complexes were there. Do you remember? Just we discussed the photosystem 2, then that photosystem 2 will pass the electron to ferrodoxin, right? Then that electron will go to what? UQ, that will become UQH2, and then finally to the cytochrome B6F complex. Do you remember that? Anybody in the class? Anybody in the class? Do you remember that? Yes, that this is something that we have discussed. It was also the system of electron transport, and in that photosynthesis chapter I told you that why do we transport the electrons, right? Because this thing, right, when we move that electrons, yes my dear students, when we move that electrons, when they move, right, when these electrons, when they move, what's going to happen? What is going to happen? The energy will be produced. That energy was used, right, for the H positive movement, if you remember, right? So H positive, basically more and more H positive, more and more H positive, they were getting accumulated, right? More and more H positive, they were getting accumulated here in the lumen. More and more H positive, it was getting accumulated here in the lumen. Are you getting my point? Are you getting my point? So basically, what were we doing? We were making the proton gradient. What were we doing? We were making the proton gradient, right? What were we doing? We were making the proton gradient. Is that clear? Is that clear? So that proton gradient, ultimately from that CF0, CF1 particle, ATP production was there. Now, here we are talking about the respiration. We are talking about the respiration. Here we are going to talk about the mitochondria. Here we are going to talk about the mitochondria. Are you getting my point? here, what are we going to talk about? We are going to talk about the mitochondria. So you know that here we have the circular DNA. We have the 70S ribosome also, and we know that we have the cristae here. What do we have here? This cristae, and this cristae here is all enfoldings these are, and this is the oxysome that we have, right? And this is the matrix that we have, right? The oxysome is there, the matrix is there. That's what we have, guys. That is what we have. Now when you talk about this inner membrane of the mitochondria, matlab overall this is also the inner membrane. I'm not specifying ki here there will be the complexes and here not. No, overall in the inner membrane of the mitochondria, it is a membrane. It is a membrane. Definitely it is having the phospholipid bilayer, right? Definitely it is having the phospholipid bilayer. Here also in the membrane, bache, what will you see? Here also in the membrane you are going to see some protein complexes. What are you people going to see? You people are going to see some protein complexes here in the membrane, right? You people are going to see the protein complexes here in the membrane itself. And when you talk about these protein complexes, when you talk about, like, what am I saying? Protein complexes, they are made up of many other proteins also, no, right? So that some complexes are present here in the inner membrane. Just let's say here you have, right, here you have these complexes, here you have these complexes. So from these complexes the electrons will move. I'm giving you the context of the topic here, and as I said, I'll keep it very simple. I'm not going to discuss the technicalities more, right? I am not going to discuss the technicalities more. I am going to keep it very simple for all of you. The reason you know that, because I have seen that students, they find it difficult, and it's not like that, guys, it is difficult. It's just that even you don't want to put efforts to understand it. Tell me, am I right? Am I right? Even you do not want to put the efforts to understand it, right? Even you do not want to put the efforts to understand it, right? To understand it, right, bache? So we know that it is the outer membrane. It is the outer membrane, right? This is the inner membrane. This is the inner membrane, and this is the intermembrane space that we have, right? Try to understand the diagram first, okay? I'll show you many diagrams to explain this, okay? Intermembrane space, right? And here, this is the matrix. So, matlab, it's like I'm taking a part, right? It is like I'm taking a part of the mitochondria, right? It is like what? It is like I'm taking a part of the mitochondria. Can you see that? Can you see that? If it is clear, do let me know in the chat section. Do let me know in the chat section. Yes, do let me know in the chat section. So what do we have here? What do we have here? Right? We have the outer membrane here, right? We have the inner membrane here. We have the intermembrane space, right? The intermembrane space here, and this is the matrix, and these enfolding, you know that they are nothing but the cristae, okay? And here in the inner membrane, we are going to discuss about the protein complexes, which are present here in the membrane itself, okay? Which are present here in the membrane itself. We are going to talk about the protein complexes, and definitely, these protein complexes, they are fixed. Please try to relate it. Try to relate it with the photosynthesis part. Kindly relate it with the photosynthesis part. I hope you remember it from the photosynthesis, right? I hope you remember it from the photosynthesis, right? So you know that there are the protein complexes, and in between there are mobile electron carriers also. In between there will be mobile electron carriers also that we need to understand, okay? And bache, this oxysomes, the F naught of that oxysome is in the matrix, right? That F naught of the oxysome is in the matrix. Are you getting my point? This F naught in the oxysome, it is in the matrix, right? So this oxysome is through which the facilitated transport of proton will be there. So ultimately, when we talk about the electron transport chain, we are going to say that basically the protein complexes are present in inner membrane of mitochondria. Right, bache? Right, bache? There are 4 complexes which are making the electron transport system, and the 5th complex is your ATP synthase. Right, and the 5th complex is your ATP synthase, and through that 5th complex, the protons will cross. Now, in the case of chloroplast, it was the thylakoid lumen in which protons were getting accumulated. It was the thylakoid lumen in which protons were accumulated. Here in electron transport chain, you people will see that protons gradient will, again proton gradient we are going to develop here, but that protons, that H positive will be accumulated here in the intermembrane space. In the intermembrane space, a very common mistake. It's a very common mistake. So let me discuss this again. So when it is a chloroplast, in the chloroplast, we talk about the thylakoids, we talk about the granum basically. So here we discuss that H positive. Here we discuss that H positive, they get accumulated in the lumen. They get accumulated in the lumen. And in the surrounding, you know that this is the stroma part. So more H positive will be there in the lumen. But in the case of mitochondria, bache, in the case of mitochondria, what you people are going to see in the case of mitochondria? In the case of mitochondria, you will see that H positive. You will see that H positive, it will get accumulated here in the intermembrane space. Right? So ultimately, here also we discuss the gradient. Here also we will discuss the gradient. Right? Here also we had discussed the gradient part. Here also we are going to discuss the gradient. Right? We are going to discuss the gradient here also. Right, bache? Right, bache? Right? So any doubt here? Any doubt here? Any doubt here? Are you sure? Yes, are you sure? Are you sure? So these are the confusion. These are the confusion that students, these are the things that students experience. Now, electron transport system. 4 complexes are the part of electron transport system. This 5th complex will help in the oxidative phosphorylation. So when we talk about these complexes, so here, again, the trick is not that good. It's NADI, SUCD. The SUCD is suicide basically. NADI, suicide, cyto 2RO. Right? I know the tricks are pathetic, but trust me, they'll help you. NADI, SUCD, it is SUCD. You can also, it's not suicide, it's SUCD. So NADI, SUCD, cyto 2RO. NADI, SUCD, cyto 2RO. NADI, SUCD, cyto 2RO. Right? NADI, SUCD, cyto 2RO. So basically, as I said, what do we have here? We have 4 complexes. NADI means NADH. NA means NADH. D means dehydrogenase. So the very first complex, bache, it is the NADH dehydrogenase complex. It is the NADH dehydrogenase complex. SUCD, right? Remember the enzyme of Krebs cycle? Succinate dehydrogenase. Succinate dehydrogenase. Right, bache? Then comes the complex number 3. Then comes the complex number 3, cyto 2RO. It is cytochrome. What is it? It is cyto 2, means 2 times cytochrome. RO. Pick up the first R. This complex is the cytochrome reductase complex. This complex is the cytochrome reductase complex. And it is RO. So this complex number 4 is the cytochrome oxidase complex. What is it? It is the cytochrome oxidase complex. What is it? It is the cytochrome oxidase complex. So NADI, NADH dehydrogenase complex. SUCD, succinate dehydrogenase. Then cyto RO, cytochrome 2 times. R means reductase. O means oxidase. These are the name of the complexes. right? And you should definitely know this, right? These are the name of the complexes. Please write it. Yes, please write it. Please, please, please. Please do it. Right? Done, bache? Name of the complex is clear. Name of the complex is clear. C reductase complex, cytochrome reductase complex, cytochrome oxidase complex. So this cytochrome reductase, so again I'm writing the names here. Complex 1, complex 2, complex 3, and then we have complex number 4. Right, complex number 4. Is that clear? Is that clear? So when it is the complex number 1, so it is NADH dehydrogenase. It is NADH dehydrogenase complex. Complex number 2, succinate dehydrogenase complex. Right? Complex succinate dehydrogenase complex. Third one is cytochrome reductase complex. It is also referred to as cytochrome C reductase complex. And bache, there is another name of this complex that we should know, that is cytochrome BC1 complex. I thought that firstly you should remember this, then I will tell you about this one. And this one is simple. It is cytochrome oxidase complex. Cytochrome oxidase complex. Clear? Clear? So NADH dehydrogenase, succinate dehydrogenase, cytochrome C reductase complex, cytochrome BC1 complex, cytochrome C oxidase complex is there. Right? Cytochrome C oxidase complex is there. Is that clear? Complex 1, 2, 3 and 4. Complex 1, 2, 3 and 4. Complex 1, 2, 3 and 4. So sometimes they can even ask you the name of complexes. Right? Name of complexes. Okay, so these 4 complexes are the part of electron transport system, right? Chaliye, देखो, we have NADH, we have FADH2. Actually, बच्चे, they will transfer their electrons. Electron transport system, when we define it, we talk about the electron and the hydrogen carriers that are present in the electron inner membrane of the mitochondria. What do we discuss? We talk about the electron and the hydrogen carrier that are present in the inner membrane of the mitochondria, right? That are present in the inner membrane of the mitochondria. Are you getting my point? So NADH2 and FADH2 is the thing. NADH2 and FADH2 is the thing. They need to pass their electrons. One thing that you need to remember, okay? One thing that should be clear in your mind. When you talk about this NADH2, it has to pass its electron. There are some routes that they follow. There are 2 routes of electron flow. Just sometimes, let's say if you need to travel from Kerala to Himachal Pradesh, there is a route that you have to follow. There is a route you have to follow. Like let's say you have to take the flight or train or whatever it is, you will check, okay, that is how I can go, right? In this way it will be cheaper for me or something. You will plan accordingly, right? Let's say if I have to go to somewhere, right? Let's say even from, right now I'm in Noida. So if I have to go to Himachal, so accordingly I'll see that what is feasible for me to drive the car or the train or the bus or whatever it is. Right? And obviously there is a way that I have to follow. It's not like that ki I am going to the Kerala and I'm thinking that, okay, from Kerala then I will go to the Himachal. Of course not. Accordingly we plan. Accordingly, right? You know, there is a way, there is a route that we have to follow. Same is the case here, my dear students. Right? So there are routes of the electron flow as well. Like when you are talking about, this is something that you need to keep in your mind. Right? This is something that you are going to need to keep in your mind. When we talk about this topic, there are so many things that we discuss. Okay? But as I said, I'll keep it very simple. I only want to discuss the points from where the question can come in paper. I am not going to explain the logic behind as well. I'm making it very clear because I have seen students struggling in this topic. But I want that if you, like, you should know at least that much. On the basis of that, you know, I want you people to answer the question. I don't want that you people are like, "Arey yaar, it is from electron transport chain. I'm missing it." No, No, you should have that confidence कि यार I can answer the question on the basis of that also. So there are roots. So when you talk about the root number 1, in the root number 1, it is the NADH that will pass your electrons. It will pass the electrons to complex number 1. Then complex number 1 will carry that electrons to complex number 3. And then that complex number 3 will carry that electrons to complex number 4. And we know that finally there is the oxygen, which is the terminal acceptor. Right? Finally, they'll go to the oxygen. Okay? So what is the root of the electron flow from NADH? It will firstly pass to the complex 1, then to complex 3, then to complex 4. Finally, oxygen. When you talk about the FADH2, second root I'm talking about, it will pass its electrons to complex number 2, complex number 3, complex number 4. Right, बच्चे? Right? So NADH 2, complex 1 to 3 and then to 4. 2, 3, 4. Is that clear? And somewhere it is making sense also. If you remember this FADH2, when will that FADH2 form? Remember in the Krebs cycle, when your succinate, when it formed fumarate, you know that your complex 2 catalyzes that reaction. The succinate dehydrogenase. What is your complex 2? It is the succinate dehydrogenase. Remember, it is the enzyme of Krebs cycle which is there in the inner mitochondrial membrane. So obviously that is why it is giving its electron to complex 2, then to 3 and then to 4. This root thing clear? Anji, Sony, this root thing clear? Okay, what is the meaning of the electron flow here? Now, they are fixed. These complexes, these protein complexes having many other things, they are fixed to a place. Just say here in the membrane 1, 3 and 4. Then obviously we should have something in between, the mobile electron carrier, movable electron carrier. There should be movable electron carrier. Just say, right? There should be movable electron carrier. Obviously quinone. We are going to talk about the quinone. And here, what will you discuss, bache? Here, you are going to talk about your CQ. What are we going to discuss? Sorry, UQ. What are we going to discuss here? UQ. Right? UQ. Just in the chloroplast, we talk about the PQ. Here, we will talk about the UQ. Right? We will talk about the UQ. Yes. So, Q basically is quinone. Q basically is quinone. Right? Right? So, UQ, right? That is ubiquinone. And sometimes we can also write it as COQ, मतलब coenzyme Q. You can call it ubiquinone, you can call it coenzyme Q. Okay? You can call it ubiquinone, you can call it coenzyme Q. Firstly, tell me this part is clear or not. Right? The Q is present there. You are talking about the quinone here. You're talking about the quinone here, right? So it is the ubiquinone, or we can also call it as coenzyme Q, right? Whatever it is. So in the NCERT, UQ is given, we go for that. So these are the electron carriers which are present in between. Just say, again, I'll write it here. Right? So complex 1, complex 3, complex 4, and here in between there should be the mobile. It is making sense. If you know the topic of photosynthesis, then definitely you will be able to relate it. Mobile electron carriers, movable electron carriers that can carry the electron from 1 complex to 3. So as I said, you have UQ here. This is the ubiquinone. Right? And we also talk about, we can also call it as COQ, that is your coenzyme Q. Am I clear here? coenzyme Q. Now complex 3 to complex 4. Again you have cytochrome C here. It is the mobile electron carrier, movable electron carrier. Okay? And as I said, the terminal electron acceptor is your oxygen. Understood? It is your oxygen. Now when it is FADH2, it will pass its electrons to complex number 2, succinate dehydrogenase complex. Again, it has to go to complex number 3, then to complex number 4. And finally to the oxygen. Am I right? Am I right here? So again here the story is same. Even complex from complex 2 to complex 3, who is going to carry the electron? Your ubiquinone. Here it is cytochrome C, mobile electron carriers. The story is same. So in the paper they can also ask you, FADH2 is going to give its electrons to complex 2, then 3, then 4. Is that clear? Now when you talk about these complexes, they are also made up of something, right? They are also made up of something. Matlab, they are complexes of course, right? So they are also made up of something. Jaise NADH dehydrogenase. Bache, it contains FMN and FES. Do you know what's that? Do you know what is that? It contains FMN and FES. It contains FMN and FES. That is flavin mononucleotide and iron sulfur centers. Okay? That will help. That will take part in the electron transport process. One single FMN means flavin mononucleotide and 6 to 7 iron sulfur centers. Iron sulfur centers. Is that clear? Is that clear? Even if it is not clear, we have to remember this. Then succinate dehydrogenase. Succinate dehydrogenase. It is having FAD or FES. So when it is FAD, what is it? FAD is what? FAD is what? You know that? Yes, bache. FAD, flavin. What's that? Flavin adenine dinucleotide. Right? What is this FAD? What is this FAD? Flavin adenine dinucleotide. Right? Just say flavin mononucleotide FMN. But here it is flavin adenine dinucleotide. Right? This is making this complex. Now third is cytochrome BC1 complex, or we can also call it as cytochrome C reductase complex. The same thing. So here it is B. It's like that. BFESC1. BFESC1. What's that? BFESC1. Is that clear? Is that clear? BFESC1. Yes? B type cytochrome, then FES, then C1. Done? Done. Then there is the cytochrome oxidase complex. Bache, it is very interesting. It is having the copper centers also. Right? It is very interesting. It is having the copper centers also. Okay? What does it contain? It contains the copper centers. But here you will talk about the cytochrome A and A3. Right? Cytochrome A and A3. Right? Just here it is made up of what? It is having cytochrome A and cytochrome A3. So that is also a PYQ. So in this particular complex, you will see two copper centers. Two copper centers. Right? What are you going to see? Two copper centers. Not the iron sulfur center, but the copper centers. Not the iron sulfur centers, but the copper centers. Is that clear? Is that clear? Right? Here you will see two heme groups and three copper ions. And three copper ions which are arranged as two copper centers. Is that clear? Is that clear? So you will, so here the electron flow is actually like this. So copper centers are there na, cytochrome say, firstly copper center, then A, yes, when there is a flow of electrons, it will move from, yes, it will move from copper ion to cytochrome A to second copper center to cytochrome A3. So remember, first it is A, then it is A3. I know at this point of time you are thinking that ma'am it is difficult, but what you have to focus on the complexes, okay? And then what these complexes are containing, FMN, FES, FADS, BSC1 and A and A3. And this question is important, that which center contains the copper centers? It's your fourth complex. Which complex carries the copper center? It is your fourth complex. Okay? Now I'll repeat something. I'm talking about the route 1 of electron flow. To understand this thing? I'm sure you are not. Are you able to understand this thing? I'm sure. Arey yaar. I'm sure you are not. Hanji, tell me. It's O2 negative, like this I have to write, we have to write. Okay? So, see, this is the route 1 of electron flow. What's that? It is the route 1 of electron flow. Please pay attention. Route 1 of electron flow it is. Okay? Right? So, I am saying NADH will give its electron to FMN and FES. Don't you think it is your complex number 1? Then ubiquinone is in between. And then these three are making your complex number 2. Then cytochrome is seen is in between. This is making your this is making complex 3 and complex 4. ये देखो. There is no rocket science. I'm telling you. There is no rocket science here. This is complex 1. This is complex 3. This is complex 4. You understand this thing first. You understand diagram first. Then everything will be easy. Trust me, then everything will be easy. Complex 1, 3, 4. 1, 3, 4. Now let's talk about the route number 2. This. Now tell me. So if roots are clear, trust me, everything will be easy. 2, then 3, then 4. Right? Yes. So this one, that mobile electron carrier in between. Tell me, doubt? Yes, trust me, after it you will be able to answer all the questions. Tell me if there is any doubt here. That's how the electrons are going to move. That's what I'm saying. Yes? Yes? Done? Sure? Yes, that is how the electrons are going to flow. Okay, chaliye. Now what's the point here? I'll tell you. But the point here is very simple. We are talking about the electron flow. You know that when we oxidize the glucose, during this glucose oxidation, so ultimately what am I saying? I'm saying that when we oxidize the glucose, right, during glucose oxidation, energy will be, right, we have to trap the energy. Ultimately, what's our motto here? What is our motto here? We have to trap the energy. Right, bache? Right. So most of the free energy that is released during the oxidation of glucose, most of the free energy that is released during the oxidation of glucose, that is retained in the form of these coenzymes, your NADH2 and FADH2. Most of the free energy that is released by the glucose oxidation, that is released during glucose oxidation, it is retained. It is retained. And how is it retained? It is retained in the form of these coenzymes. Right? They are retained in the reduced coenzymes NADH and FADH2. Right, bache? Right, bache? So ultimately, what is going to happen in electron transport chain? That electrons will be transported, right? That electrons will be released from these NADH2 and FADH2, and we have to transfer it ultimately to the oxygen. And what that oxygen is going to do with that electron? It will form the water. It will form the water. What is water? Right? It is going to form the water. Right, bache? Water. Right? Right? Is that clear? It is going to form the water. That is the point. Now, the large amount of, right? So when we oxidize here, so they need to pass the electrons to these complexes. So the large amount of free energy, when we oxidize, when we are going to oxidize this NADH2 and FADH2 here, the large amount of free energy is released. What is going to happen? They pass their electron from 1, 3 and 4, then 2, 3 and 4. So large amount of free energy is released. We are using that free energy to synthesize ATP. Up to this part all clear? Up to this part all clear? So basically, because we are oxidizing NADH2 and FADH2, right, the large amount of free energy is released. That energy is used for the synthesis of ATP, right? So that is why this is the oxidative phosphorylation. That is why complex 5 is involved in oxidative phosphorylation, because that energy is provided due to the oxidation of this NADH2 and FADH2. So this part clear? This part clear? Yes? Yes? Yes, yes, yes. Yes? Done? Okay, so one thing that you need to keep in your mind here is the electrons will transport. The electrons will transfer throughout in the electron transport chain from these complexes. Even in bacteria, even in prokaryotes, we have this electron transport system, but that will be in the plasma membrane. They don't have the mitochondria. So that will be in the plasma membrane, but they do have. So electrons will pass from complex 1 to complex 3, complex 4. But how these complexes are arranged? As per their oxidation reduction potential. Do you know that? How are they arranged? They are arranged as per their oxidation reduction potential. Can you elaborate this thing? Can you elaborate this thing? Yes. right? देखो, we are moving the electrons from these NADH or FADH to finally to the oxygen. So when they start, right, initially they have high energy. By the time they reach there, the energy will also decrease. ठीक है बच्चे, by the time they will reach here, the energy will also decrease. Please pay attention here. So where I am saying that electrons will start with very high energy and slowly they will lose it as they pass along the chain. Are you getting my point? Are you getting my point? So the complexes that are arranged here, as I said, they are as per their oxidation reduction potential. Matlab, each complex present here is having more affinity for the electron, right? Each complex, you can write it in the chain, has a greater affinity for electrons, right? Each complex in the chain has greater affinity for electrons. I'm telling you, it is as per their oxidation reduction potential. Matlab complex 1 then complex 3. That complex 3 is having more affinity for that electron than complex 1. Then complex 4 is having more affinity for that electron than complex 4. This is a very important line here. So can I say that? Tell me, can I say that here that when we move the electrons, the reduction potential, right? The reduction potential, obviously it is as per the reduction potential. Right? The reduction potential of the next complex will be more than, right, the predator one. It will be more than the predator one. Isn't it? Matlab complex 1, 3 and 4. So complex 3 is having more affinity for the electrons. It is having high oxidation reduction potential, the redox potential, right? It is having more redox potential. Next complex is having more redox potential. So in a sequence, right, in a sequential manner, the electrons will pass from one complex to another, right? Here we are not going to talk about downhill and uphill like the light reaction. Here it is going to be as simple as that, right, bache? Right? So basically when the electrons will move, so initially that electrons are having high energy and with the time gradually it will lose the energy. ठीक है बच्चे? And that energy, because we are oxidizing NADH2, we are oxidizing FADH2, the energy is released, that energy is used for the synthesis of ATP. That's why it is the oxidative phosphorylation. I believe this part is clear. That is how the things are arranged. So next thing, this is what you have to remember. So if you remember this, then this thing will be very easy. Complex 1, 2, 3, who's going to carry the electron? Your ubiquinone. Right, your ubiquinone. Complex 3 to 4, who's going to carry the electron? Cytochrome 4. Complex 2 to 3, who's going to carry the electron? Ubiquinone. 3 to 4, cytochrome C. So ultimately that is something that we need to remember. Right? This is something that should be very clear to you. Right? It is something that should be very clear to you. Clear, bache? Clear. Right? Are you sure? So as I said, here also we will talk about the proton gradient. Chalo, let's discuss that, but with the help of these beautiful diagrams. So this is the inner mitochondrial membrane. These are the complexes here. This is the inner mitochondrial membrane. These are the complexes here. Complex 1, complex 3, complex 4. Okay, 1, 3 and 4. That is the fifth one. Okay, that is the fifth one. Is that clear? Now just pay attention. Look at this structure. I think it is the perfect diagram. Outer membrane, inner membrane, intermembrane space. Here we will see high H positive concentration and low pH. So when you see this complex 1, complex 3, 2, 3 and 4. Now what you need to observe, that is also PYQ. I'm not going to write it. I will explain it here only. So you please pay attention. You have complex 1. This is your complex 2, this is your complex 3 and complex 4. Look at the position of the complexes also. Your complex 1, it is in membrane, but your complex 2 is towards the matrix side. It is towards matrix side. Okay? Towards matrix side. Now, this ubiquinone, Q, it is in between the membrane. But when you talk about this mobile electron carrier, cytochrome C, it is towards that intermembrane space. and ultimately it is the oxygen which is the terminal acceptor, and that is going to form the hydrogen. Sorry, that is going to form the water. Is that clear? Is that clear? Sure. Hanji, is that clear? Pakka? Sure. Now, one thing, जैसे we see here कि complex 1, 3 and 4, they appear like an associated structure. कि they are all together, right? They appear to be an associated structure, and for that we use the word respirosome. Respirosome. These three complexes, they are associated in such a way, right? For all these three complexes together, we also use the word respirosome. Is that clear? We also use the word respirosome. Is that clear? Is that clear? Yes or no? Yes or no? Right, so bache, how did the electron transport start? Just say, let's say this is your NADH2, it will pass its electron. So actually from NADH2 the hydride will come out. What's the word hydride? H negative. H negative. So that hydride will taken out from NADH2, that will get converted into the proton and 2 electrons, and that's how the electrons will move from one complex to another. Now, what's the next part that we need to remember? I told you about the electron, their oxidation reduction potential as well. Ye dekho. One, it will go to this UQ. Now, this is the ubiquinone. You can relate it with the plastoquinone also. You remember when you talk about the ubiquinone, UQ, it is going to accept the electron from the complex 1. Don't you think it will get reduced? Sorry, it will get reduced when it will accept the electron. So this ubiquinone, it is also the hydrogen carrier. It will also make UQH2. So it will also pick up the hydrogen from the matrix side, and when, right? So here, right, just like your Z scheme, UQ will form UQH2. Means ubiquinone forms ubiquinol. So what is this ubiquinol? It is the reduced ubiquinone. So obviously this one will also pick up the H positive from the matrix, and when it will pass the electron to the third complex, it will also give the H positive to the intermembrane space, because we have to generate the proton gradient here, right? So a Q cycle works in them, right? A Q cycle is going on here, and again we are not talking about it, because if I will go here and there, you are not going to understand it. Right, so I'm not touching that technicalities here, but I want you people to remember this, because it is just like your, right, it is just like your what? It is just like your, yes bache, that Z scheme part. Okay, it is just like your Z scheme part. So what is going to happen? Electrons are transferred along the electron transport chain by the reversible reduction and oxidation, right? So yes, yes, this part clear? So complex 1, it is having FMN, FES, you have to remember this. So ultimately the electrons will move. Now, one thing that you need to remember, yes, one thing that you need to remember, okay, come to this part of the NCERT, right? With time you will remember this. Ye dekho. This NCERT diagram, I am elaborating it. Bache, see, I am drawing the diagrams like this, but they are showing it in this vertical way, right? So here rest things are same. Just complex number 1, then they are showing complex number 3, then they are showing your complex number 4, and then they are showing your, then they are showing your complex number 2, and then they are showing the complex number 4 and 5th. But you have to consider the diagrams that I am showing you. Why? Because they are telling you the position as well. Ki succinate dehydrogenase is towards the matrix side, cytochrome C is towards the intermembrane space, ubiquinone is somewhere in the traversing in the membrane. So we should know that things. Okay? Now here matrix, here intermembrane space, right? I think this portion is clear to you. Right, I think this part is clear to you. Now, even in the diagram, what are they showing? 2 electrons are coming out. I told you how the process will start. A hydride be taken out from NADH2, hydride, matlab H negative. So it will form 2 electrons and proton, right? And then that 2 electrons will move. But no need to focus there. No need to focus there. Just focus on the questions that are going to come in your paper. That electrons, they will go to FMN, they will go to FES. If you will observe the diagram, here it is 2 electrons, here it is 1. As I said, I'm not going to discuss that things in detail, right? That why this happened, how this is going to happen. I will just tell you few points that you need to remember. Okay? Now, this is complex 1. Your electrons will pass to ubiquinone. That will form UQH2, right? You know that it will pick up the H positive from the matrix also. Now it will pass its electron to cytochrome B, because complex 3 is made up of cytochrome B, FES, C1, and that will pass the electron to cytochrome C. That will carry it to the complex number 4 having copper A center, cytochrome A, cytochrome A3, and then copper B. That is what we have here. Yes, that is what we have here. That is what we have here. But if you talk about the concept, that is how we draw it. Firstly, we write copper center A, cytochrome A, then cytochrome A3 is there, but firstly copper center B will come. Okay, what will come? Copper center B will come, then it will go to the cytochrome A. Matlab NCERT is showing like this na, but I'll tell you, just copper center A, cytochrome A, copper center B, cytochrome A3, like this it should be, like this it should be. And finally that electrons will go to the oxygen, right? Finally that electrons will go to the oxygen. Now look at this. Your complex 1 is pumping 4H positive. Matlab, when the electrons get transferred from this, complex 1 pumps 4H positive, right? Your complex 3 also pumps 4H positive, but your complex 4 pumps 2H positive. That's something that you need to remember. I'm again telling you, I'm making it very clear there is a reason behind. I'm not discussing that reason right now. That reason, nobody is going to ask you that reason in your paper, right? And if I will start explaining the reason, again you people will get confused and you will leave this topic, and I don't want that, right? So I am telling you the things that you need to remember. Where the ETC will occur? Inner mitochondrial membrane. How many complexes are there? 4. Right, NADH will pass the electron to complex 1, then 3, then 4. In between 1 and 3, you have ubiquinone. In between 3 and 4, you have cytochrome C. In between, FADH will pass the electron to complex 2, then 3, then 4. 2 to 3 in between ubiquinone, 3 to 4 in between cytochrome C. Ultimate acceptor is oxygen. That is what I just said. Now, then I told you about the positions. Then I said complex 1 is having FMN and FES. Complex 2 is having FAD, FES. Complex 3 is having cytochrome B, iron sulfur center, cytochrome C1, then cytochrome C. Complex 4 is having 2 copper centers, copper center A and B, and A and A3. So copper A, then cytochrome A, then copper center B, then cytochrome A3. That is how it is. Is that clear? Right? Is that clear? Okay. Now complex 1 from the matrix in the intermembrane space, it is pumping 4H positive. Complex 3 is also pumping 4H positive. But complex 2, when it comes to complex 2, it is not pumping any proton. You know, the reason can also be it is towards the matrix side. That's why it cannot pump. We are not talking about it. But your complex 2 will pump 2H positive. Why? Because that extra 2H positive will be accepted by your oxygen so that it can form the water. Just here, dekho. Here they have taken half oxygen. When it is half O2 plus 2H positive, you will get 1H2. If it is oxygen plus 4H positive, then you will get 2H2. Okay? Then you will get 2H2O. That's how you have to remember. So why this complex 4 is throwing out 2H positive in the intermembrane space? Because that extra 2H positive from the matrix will be accepted by half oxygen so that it can form the water. But complex 2 is not pumping any proton in the intermembrane space. It is not pumping any proton in the intermembrane space. Okay? It is not pumping any proton in the intermembrane space. So matrix inner membrane, intermembrane space. ठीक है, we are talking about NaDH2 here. Here we are talking about FADH2. Again, matrix, inner membrane, intermembrane space. Complex 2, complex 3, complex 4. Complex 1, complex 3, I am not specifying the position yet. Complex 4. This one is pumping 4H positive from matrix to this. This one is also pumping, but this one is pumping 2H positive. Why? Because 2H positive will be accepted by half oxygen, that will make H2O. Now here, this one will pump 4H positive, this one will pump 2H positive. So when we are talking about the transport of electrons, transfer of electrons from NaDH2, can I say that 10H positive, 10H positive will be there in the intermembrane space? Can I say that 10H positive will be there in the intermembrane space? Can I say that 10H positive will be there in the intermembrane space? Yes? Yes? And here it is just 6H positive, right? It is just 6H positive, right? It is just 6H positive, right? Now, again, these protons, they cannot, these protons, you know that they cannot cross the membrane directly. These protons, they cannot cross the membrane directly. You know that we have more and more and more and more H positive here in the intermembrane space. What is it? It is the intermembrane space, and we have more H positive here. So these H positive, they will move by a facilitated diffusion. How will they move? They will move by a facilitated diffusion. What is the meaning of facilitated diffusion? Which diffusion means things will move along the concentration gradient. What is the meaning of this diffusion? Things will move along the concentration gradient, down the concentration gradient, from high concentration to the low concentration they move. But it is the facilitated diffusion, means some things they cannot cross the plasma membrane directly. They need a channel through which they can pass. Right, they need the channel through which they can pass. So when you are talking about that channel, it is your oxysomes or ATP synthase. So your H positive through this fifth complex, right, because this, we know that this electron transport will generate the energy, right? The electron transfer will generate the energy, and that energy is required for the, that energy is used for the synthesis of ATP. That's why it is oxidative phosphorylation. So what is going to happen? These protons are present here. So these protons from intermembrane space through this oxysome, right? Because here their concentration is more, in the matrix their concentration is less. So they will move along the concentration gradient. They'll come along the concentration gradient, they will form the ATP here, right? This energy is used for the formation of ATP. So what is the point that you need to remember here? The point here that you need to remember is that when 4H positive cross this oxysome, 1 ATP is formed. When 4H positive, they cross the oxysome, when 4H positive, they cross the, right, they cross the oxysome या ATP synthase, you will get, what will you get, right? You guys are going to get, you people are going to get 1 ATP. ठीक है, 1 ATP, it is the 4H positive. So in your NCERT, in this diagram, they are mentioning 2H positive. No, it will be 4H positive. So 4H positive, when they cross this, 1 ATP will be formed. Now imagine, now you imagine, ठीक है, NADH, because of NADH, total 10H positive are there in intermembrane space. They are in intermembrane space. Yes, they are in intermembrane space. So 4H positive will give you 1 ATP. Next 4H positive will give you one more ATP, and then 2H positive will give you what? Half ATP, 0.5 ATP. That's why we say that 1 NADH gives us 2.5 ATP, right? 2.5 ATP. 1 NADH gives us 2.5 ATP. Now, your FADH2, 6H positive are there in intermembrane space. So 4H positive will give 1 ATP and 2H positive will give 0.5 ATP. That is why we say 1 FADH2 gives us 1.5 ATP. So earlier, last year, it was mentioned as 3, it was mentioned as 2, but exactly it is 2.5 and 1.5. So we have to follow the concept. We have to follow the concept. Is that clear? We have to follow the right? We have to follow the this concept here. Is that clear? Is that clear? Done? Sure. So now let's talk about it. See, in the link reaction, okay, 2 pyruvate are giving us 2 acetyl coenzyme. 2 pyruvate are giving us 2 acetyl coenzyme. And here we are getting 2 NADH2. Okay? And these 2 acetyl coenzyme A in Krebs cycle, they are giving us 6 NADH, 2 FADH2 and 2 ATP. Am I right? Am I right? So let's discuss that. Here 2 multiply 2.5, 6 multiply 2.5, 2 multiply 1.5. That's it. Means 2 NADH2, they will give us how many ATP? Yes, they'll give us how many ATP? Tell me, they'll give us how many ATP? Now you have to consider 2.5 only. Right? You have to consider 2.5 only. They'll give us how many ATP? Obviously, 5 ATP here, 5 ATP here, and here it will be, please tell me, here it will be 6 into 2.5, 6 into 2.5, 6 into 2.5. Right, and here it will be 3 ATP, isn't it? Isn't it? So from the link reaction and from the Krebs cycle, in totality, from the link reaction and from the Krebs cycle, in totality, you are getting 25 ATP. Take it, 2 ATP plus 3 plus 15 and then 5. Am I right? Yes. Am I right? Am I right? Done? So, see, as per old NCERT, it will be 3, it will be 3, it will be 2. So, as per old NCERT, here it should be. As per old NCERT, it is 30. As per old NCERT, it is 30. Okay, it is 30. But obviously, we have to follow the concept, and that is saying 25 ATP. Okay? Done. Okay? And this is from the link reaction and Krebs cycle. This is from the link reaction and Krebs cycle. I'm not talking about the glycolysis here. I'm not talking about the glycolysis here. Now let's consider the glycolysis. You know that from glycolysis also we get 2 NADH2. We get 2 NADH2. Now, bache, it is in the cytoplasm. It is in the cytoplasm. If we want the ATP from it, we have to send it in the mitochondria, right? We have to send it in the mitochondria if we want ATP from it. If we want ATP from it, what we have to do? What we have to do? From cytoplasm, we have to carry it to the inner membrane of the mitochondria. That is not possible directly. NADH2 cannot cross it directly. So here comes some shuttle system. Here we talk about the shuttle system. That shuttle system will help, right? With the help of that shuttle system, electrons from NADH2 will pass to the electron transport system. So there are two shuttle systems that we discuss, bache. One is your malate aspartate shuttle system. One is your malate aspartate shuttle system, and second is, anybody in the class who can answer this? One is malate aspartate shuttle system, and second system is, anybody in the class? Anybody in the class? Malate aspartate shuttle system? Glycerol phosphate shuttle system. Glycerol phosphate shuttle system. What's that? It is glycerol phosphate shuttle system. There are two shuttle systems which will carry these electrons to that inner mitochondrial membrane. Right, bache? So in the malate aspartate shuttle system, the electrons from NADH2 in cytoplasm, electrons transfer from NADH2 in cytoplasm to NAD positive in the inside mitochondria. Matlab, the NADH2 which is there in the cytoplasm, it has to pass its electron because it also wants to form the ATP. So if it will give its electron, matlab if it is following this malate aspartate shuttle system, so it means it will transfer its electron to oxidized NAD which is inside mitochondria. So if oxidized NAD, if it will get electron, definitely it will give it to complex 1, then 3 and then 4. So here also we will get how many ATPs? Here also we will get how many ATPs? You know that we are going to get 2.5 ATP, right? From 1 NADH2, 2.5 ATP, 2 are there, 2 NADH2 are there. So we will get, yes, 5 ATP, yes or no? We will get, from 1 we are getting 2.5, so from 2 we will get 5 ATP. Now there is one more thing, that is glycerol phosphate shuttle system. There is one more thing, that is glycerol phosphate shuttle system, right? So in this shuttle system, electron transfer occurs from means it will give its electrons to FAD. Okay, it will give its electrons to FAD inside mitochondria. So you know that FAD, 1 FADH2 gives us 1.5 ATP. So here, if your NADH2 will give its electrons to FADH2, so FAD, then here from what will you get? You will get 3 ATP only, and here you will get one more ATP, that is 5 ATP. So that is also that which shuttle system, right? Which shuttle system is carrying the electron? Which shuttle system is carrying the electron? That will also decide the number of electrons. ठीक है बच्चे? That also decide the number of electron. Is that clear? Is that clear? Sure? Sure? Done. So this malate aspartate shuttle system, it operates in kidney, heart, liver. This one, it operates in kidney, heart, liver. More energy is required, of course, right? And the another one will be there in the muscle cells, right? and the nerve cell. This one, it predominates in eukaryotes. It's predominant in eukaryotes. And it operates in, examples are your muscle cells and the nerve cells. Okay? So this is about the shuttle systems also. Right? So that is why when we talk about the glycolysis, because in glycolysis you are getting 2 ATP, 2 NADH2. So obviously old NCERT was saying 2 into 3, 6 ATP here, 2 ATP here. So old NCERT was saying a total 8 ATP. That's why when we solve the questions, that is why when we solve the questions, we used to see this thing. Okay, from the glycolysis, the total number of ATP after complete oxidation of glucose will be 8. But this is inappropriate. The actual concept is here at the place of 3, it is 2.5, right? So it will be 5 ATP. So it will be 5 plus 2, it should be 7 ATP. So in the paper, in the final paper, in the NEET paper, you have to see the option and you have to act accordingly. Let's say if in the option 8 and 7, both are given. So obviously you have to select the actual answer, because then you can challenge it, right? Then you can challenge it. And right now, obviously, I'm not considering the shuttle system ka complex here. So that is how we have to calculate the total number of ATP. So see, malate aspartate shuttle system. This is the cytosol intermembrane space. So what is happening? What is happening? It will pass its electrons to this, right? NADH, right? It will pass its electrons to NADH. And then finally it will go there. Bas. That's how you have to remember. And this is the glycerol phosphate shuttle system. So it will pass the electrons to this FAD. Okay. So accordingly the things will change. So this is all about this complete oxidation of glucose. And now we have to discuss this part, the respiratory balance sheet, amphibolic pathway, and RQ value. All the topics are important. So I need your 30 or 40 minutes and we will finish the chapter. Okay? I need your 30 to 40 minutes and we will be able to finish the chapter. Right, bache? Right? Okay? Chaliye. So let's continue this. Now let's read the ETC. Let's read the ETC from the NCERT, and I hope now you are able to understand it as well. So electron transport system and oxidative phosphorylation. So remember, when it is ETS, complex 1 to 4 are involved. When it is oxidative phosphorylation, your complex 5. So when they are asking both ETS plus oxidative phosphorylation, both, all the 5 complexes are there, but here only first 4. So what are they saying? The following step in the respiratory process are to release and utilize the energy stored in this and this. Right, so when they are oxidized through the electron transport system and the electrons are passed on to the oxygen, this will result in the formation of H2O. So ultimately what is going to form here? Ultimately what is going to form here? The H2O. Okay, the H2O. So metabolic pathway through which the electron passes from one carrier to another is the electron transport system, and it is present in the inner mitochondrial membrane. So electrons from NADH produced in the mitochondrial matrix, they during citric acid cycle are also oxidized by complex number 1. So electrons are transferred to ubiquinone located within membrane. It also reduces FADH2, right bache? So reduced ubiquinone is known as ubiquinol. Again, very important thing. It will oxidize with the transfer of electrons to cytochrome C by a cytochrome BC1 complex, and then cytochrome C is a small protein attached to the outer surface of inner membrane. Matlab it is towards intermembrane space. towards that side. Okay, so it is the mobile electron carrier, movable electron carrier for the transport of electrons. Okay, so complex 4 refers to the cytochrome C oxidase complex containing cytochrome AA3 and 2 copper centers. And 2 copper centers. Is that clear? Is that clear? Right, bache? Is that clear? Sure? Sure? Are you sure? So when the electrons passes from one carrier to another, right? So they are coupled to ATP synthase. That will help in the ATP production. So cytochrome C, as per NCERT, that is your mobile electron carrier. That is your movable electron carrier. Is that clear? Is that clear? Sure? Right? They are your mobile electron carrier. Right? These both are basically your mobile electron carrier. Okay, mobile means movable. It's not like that they are having the mobile phone. So oxidation of one molecule, again NCERT is saying 3 molecules of ATP, but actually it is 2.5. 2.5. Okay, so you have to remember that. You have to remember that. Okay, so the presence of oxygen is vital. It drives the whole process by removing hydrogen from the system. So oxygen is the final terminal acceptor. It is the final hydrogen acceptor. It is the final hydrogen acceptor. So unlike photophosphorylation, here it is not the light energy that is making the phosphate, but in respiration, it is the energy of oxidation reduction which is used for the formation of ATP. That's why it is oxidative phosphorylation. Okay, bache? Okay, bache? Right? So we know about the chemoosmotic hypothesis. So accordingly, here also we need to understand that how that, protons will move. So look at this diagram. So here you can see the Krebs cycle, FAD, FADH2, they will pass the electron to the second complex, then this will pass the electron to this ubiquinone, then complex 3 and then to 4. Okay, so that is how it is. So ultimately H positive will be sent here in the intermembrane space. So you have to remember complex 1 will pump 4H positive, complex 3 will pump 4H positive, 4 will pump only 2, and complex 2 will not pump any H positive. That's something you need to remember. I'm not going to explain why and why not. Okay? Okay? Done? Okay. ठीक है? चलिए, so just look at this enzyme, right? Just look at this diagram. I hope it is clear, and if it is not, do ask me the questions. Yes, you can ask me the questions here if it is not clear. Bache, please tell me if there is any doubt. Please tell me. Any doubt? Yes? Any doubt? Sure? Okay. So now the respiratory balance sheet. Okay, here bache, there are some assumptions also. Again, question can also come from assumptions, right? Question can also come from assumptions, right bache? Right, chaliye, let's discuss that assumption. Just a respiratory balance sheet. It is possible to make calculation of net gain of ATP for every possible molecule oxidized, but in reality, this is just a theoretical exercise. Matlab, we say that number of ATP will be produced by Krebs cycle, that number of ATP will be produced by glycolysis. Actually, it is something theoretical. Do you think in the body things are going to work in such an orderly sequence? In such an order? Do you think so? Matlab, we are saying that glucose will form glucose 6-phosphate, then this and that. We don't know the cellular need. Maybe there is a need in which, and might be your alpha ketoglutarate of Krebs cycle, maybe it get converted into something else, right? Maybe the fatty acids, they'll get converted into something else. We cannot say that. So basically there are some assumptions. That assumptions are, we are assuming this, ki there is a sequential orderly pathway functioning with one substrate forming the next and with the glycolysis. So what are they saying? They are saying that there is a proper orderly pathway which is functioning with one substrate forming the next and with glycolysis, TCA and ETS falling one after another. So we are assuming that one metabolite, one substrate is forming the next, and we are also assuming that after glycolysis there is TCA, there is ETS. So we are assuming that pyruvate is entering in the TCA only, right, and then it is going in for the ETS only. That's what we are assuming. So NADH synthesized in glycolysis is transferred into the mitochondria and undergoes oxidative phosphorylation. Again, this is also our assumption. We are assuming that. And then none of the intermediates are used in any other pathway. Matlab, they are going like that, right? They are going like this, right? The way we are assuming. It's not like that if in body pyruvate is required for something else, it is going there. No, it is moving like this only. That's what we have assumed. So only glucose is being respired. No other alternative substrates are entering in the pathway, right? That is what we are assuming. Okay, so but this kind of assumptions are not really valid in the living system, because we know that all that things are working simultaneously, right? It's not like first thing first and then second then third. No, all of it is working all together, right bache? Right, so substrates, they are entering the pathway, they are withdrawn from it. It is all working as per the necessity, as per the need, right? So ATP is utilized as and when needed. Enzymatic rates are controlled by multiple means. So yet it is useful to do this exercise to appreciate the beauty and the efficiency of the living system. Here there can be a net gain of 38 ATP molecules during aerobic respiration. Now again, I know you guys are going to raise the question, how come 38? Dekho, 25 from what? 25 from Krebs cycle and your link reaction, 7 ATP from that glycolysis, so 32 ATP. Now it is as per the actual concept. If I go for that old NCERT thing, matlab it is also given in new NCERT also, right? So we can say ki Krebs cycle only. We can say that Krebs cycle only. Just say, where is it? Yes, as per that, ye dekho. Here we are making 25 ATP. As per that, 6 plus 18 plus 4 plus 2. 30. So as per that we are getting 30 ATP from link reaction plus TCA cycle and 8 ATP from glycolysis. So that is why the 38 is given here. So now you have to answer it, right, as per the options in your exam. Okay. I have explained you this thing. Now, fermentation is already compared, but chalo, let's discuss it. Ki fermentation accounts for only a partial breakdown of glucose. Okay, it's not the complete breakdown. So in fermentation, there is a net gain of only 2 molecules of ATP, and that is from the that is from the glycolysis. Even in bache, your RBCs and even your muscles, let me tell you, even your RBCs and your muscles, and especially your RBCs are also getting the energy from the glycolysis only. They don't have mitochondria, you know it, right? They don't have mitochondria, right? But just Sunny, I have explained it na, that why, how come 38, how come 32, okay? So that's it. So NADH is oxidized to NAD rather slowly in fermentation, but in aerobic respiration, this is very vigorous reaction, okay? Chaliye. So now, on what basis we are saying amphibolic? But when we are using the word amphibolic, we are including both. Amphi means dual, right? We are including both. We are saying the catabolism is there and the anabolism is there. Matlab, breakdown is there and the buildup is also there. Breakdown is there and then the buildup is also there. This is what we have. This is what we have. So here, right, who's going to explain this amphibolic pathway thing? This particular table. And it is also a previous year question. They can give you this, they can ask you what is present here, what is present here, what is present here. They can ask you anything. So you know that fats, if you know anything from the biomolecules, you know that your fats, they are made up of glycerols and fatty acids. Glycerol and fatty acids, when they join with the ester bond, they will form your lipids. So obviously, if you are breaking fats, you will get fatty acids and glycerol. Now this glycerol can form dihydroxyacetone phosphate as well, right? Yes, this glycerol can form that. So here there is a breakdown. It is getting converted into another that can form glyceraldehyde 3-phosphate. So in this way, it can enter here in the glycolysis. Your fatty acids, from fatty acids, that is also a question, separate question. From fatty acids, you guys can get acetyl coenzyme A. And you know that, then it can enter in the Krebs cycle. So that's how fats, they have their way to enter in this glycolysis and the TCA cycle, right? So we are to just assuming na ki things are going like this, then first, second, third, but that is how it is working. And then when you talk about the amino acids, that amino acids can also get converted into the pyruvic acid. So this is what you have to remember. The amino acids, they are forming pyruvic acid, and again it can enter in the process, right? So that is how things are. So what this paragraph is saying, that glucose is the favored substrate. You know that glucose, because of its RQ value, respiratory question, right? When we need, our body needs energy immediately. They say fats, they give us more energy, but still they are not our primary respiratory substrate. Why? Because for the oxidation of fat, we need more glucose. It takes time, but we need the energy immediately. If our brain cells will not get energy immediately, even the paralysis can occur, right? So all carbohydrates usually first converted to glucose, then they are used here. Other substrates are also used, but then they do not enter the respiratory pathway at the first step. Just in that figure, it is given na ki fats would need to be broken down into glycerol and fatty acids first, then they can enter. So fatty acids, I told you, right? They'll be degraded to acetyl coenzyme A, then they will enter. Glycerols will give you glyceraldehyde 3-phosphate, then they will enter. Proteins, they'll be degraded by proteases enzymes, then they'll form amino acids after deamination, right? And then they can enter in the pathway. They'll form the pyruvate and they can enter there, right? So since respiration involves breakdown of substrate, the respiratory process will be catabolic, okay? But is this understanding correct, right? Because we have seen that in respiratory pathway, different substrate would enter if they were to be respired and used to be derived energy. Right, bache? Right. So what is important to recognize is that it is these very compounds that would be withdrawn from respiratory pathway for the synthesis of the said substrate. So fatty acid would be broken down to acetyl coenzyme A before entering the respiratory pathway when it is used as a substrate. But when the organism need to synthesize fatty acid, acetyl coenzyme A will be withdrawn. Matlab, it is as per our need. If it is required, fatty acid will convert itself into the acetyl coenzyme, then it will enter the pathway. But if in our body there is a requirement of fatty acid, then acetyl coenzyme will form the fatty acid. So that is how it is, bache. That is how it is. So hence the respiratory pathway comes into the picture where breakdown and build-up both are going on, bache. That's why it is amphibolic. That's why I told you that at the end I will tell you why this is amphibolic. Okay? Now last topic here is RQ value respiratory question. So obviously oxygen is consumed, CO2 is released. So the volume of CO2 evolved to the volume of oxygen consumed is giving us RQ value, respiratory quotient value. In the case of carbon dioxide, 6 CO2 molecules are evolved, 6 oxygen molecules are used. So if in the paper it is given ki RQ value is 1, this is which respiratory substrate it is? It is the carbohydrate. It is the carbohydrate. What is it? It is the carbohydrate. Are you getting my point? Now here, look at this. See this. Now when it is tripalmitinic, that is again a PYQ. What is it? It's a fatty acid. It's a fatty acid. Here in the case of this fatty acid, 102 CO2 molecules are evolved. Oxygen used is 145. So it is 0.7. So if RQ value is given as 0.7, you are talking about the fats. You are talking about the fats. So the formula of tripalmitinic acid, please remember that. The formula of tripalmitinic acid, kindly remember this. This is important. Yes, this is important. Okay, this is important for us to remember this also. Now the next thing, let's say if it is a protein, and yes, see, this is your homework. You will also write the stearic acid formula, that is also a fatty acid, and then also you will do this reaction. Then we talk about the proteins, and in the proteins it will be 0.9, again less than 1. Okay, again less than 1. Now bache, when organic acids are the respiratory substrate, some seeds, some plants, they can use organic acids also. So when organic acids are the respiratory substrates, when they are the respiratory substrate, then what is the story? You see, you can take the example of anyone. You can take the example of oxaloacetic acid. You can take the example of malic acid. So let's take the example of oxaloacetic acid. This is the example. Do you know that? This is the example. This is your oxalic acid. Not oxaloacetate, it is oxalic acid. This is the CO2 release. Just imagine. Oxygen consumed, and look at the CO2 release. And what will be done? Water will be released. So here 4 CO2 over 1 oxygen, it will be 1. So in the case of organic acids, the RQ value is even more than 1. Right, bache? It is more than unity. Right, bache? It is more than unity. Okay, जैसे malic acid के case में it is 1.33. Okay, so here it will be more than unity. It is more than 1. Is that clear? Is that clear? Now let's say if it is in the absence of oxygen. If absence of oxygen means anaerobic conditions are there, then what will be the RQ value? What will be the RQ value? Yes, bache, what will be the RQ value? Tell me. Just say you have the example, glucose, zymase. Zymase is released from your yeast. You are talking about alcoholic fermentation. So we are getting C2H5OH, and obviously 2 carbon dioxide molecules will be released. Right, carbon dioxide will be released. So now you tell me what will be the RQ value here? What is going to be the RQ value here? What is going to be the RQ value here? Yes. So 2 CO2 and here 0, it is around infinity. So if RQ value is infinity, you are talking about the anaerobic respiration. If RQ value is more than 1, you are talking about the organic acid. If it is less than 1 and 0.9, it is protein. If it is 0.7, you are talking about the fats. You are talking about the fats. So this is all about this chapter, and do read about the floating respiration and protoplasmic respiration. Right, protoplasmic respiration, very rare it is, in which proteins, right, body will feed on proteins. Proteins are the respiratory substrate, so read about it. That's your homework. You have to read about it. So this is all about this chapter. I'll share the PDF in the Telegram group, guys. I hope now this chapter is easy for you. So you can put that in the comment section, and soon I'm going to make the video in which I will discuss all your problems that you have mentioned in one of my videos, this video, right? In this strategy video. Okay, so this strategy video, whatever problems you guys have mentioned in the comment section, I'll make a video and I'll discuss everything. Okay, then I will discuss everything. So that's it, that's all. Do read the chapter carefully. We are done with the plant physiology, and after next class we'll complete the human physiology as well. So 2 most important units are covered here in this Game of NEET 2.0 series. So next, whatever you want cell cycle, do let me know. You want that cell unit or diversity unit, whatever you need, do let me know in the comment section. Okay, so done, that's it, that's all. So we'll share the PDF, and in the Telegram group, and the link for that is also there in the description box. Okay, okay, done. And you know that it's 24th October, so at 6 p.m. you guys are going to get something very amazing, so stay tuned to that as well. And if there is any doubt, any problem, do let me know.